At this moment, the world around you may seem to arrive fully formed. There are objects with colours, sounds with locations, surfaces with textures, and a body that feels quietly present within the space it occupies. It all appears immediate, as though the mind simply opens a window and looks out at reality. But that is not quite what happens. Human beings never experience the external world directly. What reaches the nervous system is a collection of physical signals. Light enters the eyes. Pressure waves move through the air and reach the ears. Molecules drift into the nose or dissolve across the tongue. The skin changes temperature, stretches, vibrates, and sometimes suffers damage. Muscles contract, joints shift, and fluid moves through the inner ear as the head turns. From these signals, the nervous system constructs an internal model of what is happening. This model is not a miniature picture hidden inside the skull, nor is there a tiny listener seated somewhere in the brain, wearing headphones and watching the scenery. Instead, billions of nerve cells exchange electrical impulses and chemical signals across vast networks. Through that activity, the experience of a world appears. Outside the nervous system, light is electromagnetic radiation. It has wavelengths, frequencies, and energy. Yet a wavelength is not blue, red, or green in itself. Colour is the experience produced when the visual system responds to particular patterns of light. Sound begins as changes in pressure moving through air, water, or another material. Those vibrations do not carry loudness, melody, or the gentle character of a familiar voice as separate ingredients. Those qualities emerge when the auditory system analyses timing, frequency, intensity, and pattern. Food releases molecules. The nose and mouth detect them, but flavour is not simply sitting inside the food waiting to be collected. It is assembled from taste, smell, temperature, texture, irritation, memory, and expectation. Warmth begins with the movement of energy. Pain may begin with tissue damage or the threat of damage. Balance depends on gravity, acceleration, vision, muscle activity, and the inner ear. Spatial awareness depends on many signals being combined into one coherent sense of where things are and where the body is among them. This does not mean perception is unreal. A constructed experience is not necessarily a false one. A map is constructed, but it can still represent a real landscape. A weather forecast is created from measurements and models, yet it can still describe conditions outside. In much the same way, perception is produced inside the nervous system while remaining closely connected to the physical world that caused it. It is useful to distinguish between objective physical reality and subjective perceptual reality. Objective reality refers to the physical events and structures that exist independently of one particular observer. Subjective perceptual reality is the world as it is experienced by a nervous system. The two are connected, but they are not identical. The senses did not evolve to provide a complete scientific account of the universe. They evolved because organisms that could detect useful information were more likely to survive, move, feed, avoid danger, reproduce, and respond effectively to changing conditions. Human vision is therefore not a general detector of electromagnetic radiation. It is sensitive to a very narrow region called visible light, usually described as extending from roughly 400 to 700 nanometers. Beyond one edge lies ultraviolet radiation. Beyond the other lies infrared radiation. Both are real, both can carry information, and neither normally appears within human vision. Many insects can detect ultraviolet patterns that remain invisible to us. Some snakes can detect infrared radiation associated with warm bodies. Their sensory worlds contain information that ours does not. Human hearing is limited as well. A healthy young person may hear frequencies from roughly 20 to 20,000 cycles per second, although sensitivity varies and usually changes with age. Frequencies above this range are called ultrasound. Bats and dolphins use ultrasonic signals for echolocation, revealing the positions and movements of objects through echoes that human ears cannot detect. Other animals respond to magnetic fields. Migratory birds, sea turtles, fish, and some insects appear able to use information associated with Earth's magnetic field while navigating. Many species can also detect polarized light, in which the orientation of a light waves follows particular patterns. To human observers, that information is normally absent from conscious vision. The German biologist Jakob von Uexküll used the word Umwelt to describe the sensory world available to a particular organism. An Umwelt is not simply the physical environment itself, it is the portion of that environment that an animal can detect and that matters to its behaviour. A tick, a bee, a bat, and a human being may occupy the same forest while inhabiting very different perceptual worlds. The forest contains far more than any one nervous system can reveal. This is the sense in which the human senses create reality. They do not create stars, stones, oceans, or electromagnetic fields. They do not summon the universe into existence. They create experienced reality by selecting certain signals, transforming them into neural activity, and organizing that activity into colours, sounds, objects, distances, bodily feelings, and meaning. Sensory receptors begin the process, but perception is shaped by much more than raw input. Attention determines what becomes prominent. Prediction helps interpret incomplete or uncertain signals. Memory allows recognition. Emotion gives events importance. Internal bodily signals contribute to hunger, calmness, tension, and the feeling of being alive. Touch, balance, movement, and vision combine to produce the sense that the body belongs to you and occupies a particular place. What feels like a simple encounter with the world is therefore an extraordinary achievement. The brain receives fragments, comparisons, changes, and probabilities. From them it builds a stable room, a familiar voice, a warm drink, a distant sound, and a self quietly experiencing it all. Between the physical world and conscious experience, an important transformation must take place. Light, pressure, heat, chemicals, and tissue strain are all forms of physical or chemical energy. The nervous system cannot simply place these events inside the mind exactly as they are. Instead, it must convert them into a common biological language. That conversion is called sensory transduction. Sensory transduction begins in specialized receptor cells. Each type is sensitive to particular forms of stimulation. Photoreceptors in the retina respond to light. Mechanoreceptors in the skin respond to pressure, vibration, stretching, and movement. Related mechanoreceptors in the inner ear respond to sound vibrations and changes in the motion of the head. Chemoreceptors involved in smell and taste respond to molecules. Thermoreceptors respond to warming and cooling. Nociceptors respond to potentially damaging mechanical, thermal, or chemical conditions. These receptors do not carry tiny versions of the outside world into the brain. A photoreceptor does not send a miniature patch of colour along the optic nerve. An auditory nerve fibre does not contain a faint little sound. A receptor in the nose does not transport a microscopic smell toward the cortex. Instead, sensory cells change their electrical behavior. Every neuron is surrounded by a thin membrane that separates the fluid inside the cell from the fluid outside it. These fluids contain electrically charged particles called ions. Among the most important are sodium and potassium ions. The membrane controls how easily these particles can move across it through specialized protein channels. At rest, a neuron maintains an electrical difference across its membrane. The inside is generally more negative than the outside. When stimulation changes the behavior of ion channels, the electrical state of the cell begins to shift. If the change becomes large enough, the neuron may produce an action potential. An action potential is a brief traveling electrical event. Sodium channels open, allowing positively charged sodium ions to move into the neuron. This rapidly makes the inside less negative and then briefly positive. Soon afterwards, potassium channels open, allowing positively charged potassium ions to move out. The membrane then returns toward its resting condition. This electrical pulse travels along the neuron's axon without carrying a picture, sound, smell, or texture. Information is represented through patterns, including which neurons are active, how frequently they fire, when they fire, and how their activity relates to signals elsewhere. When an action potential reaches the end of an axon, it can cause chemical messengers called neurotransmitters to be released. These molecules cross a tiny gap called a synapse and influence the electrical activity of the next cell. The nervous system therefore communicates through an intricate partnership between electricity and chemistry. The 19th century German physiologist Johannes Müller recognized an important consequence of this arrangement. His doctrine of specific nerve energies proposed that the quality of a sensation depends partly on which sensory pathway is activated, rather than only on the nature of the original stimulus. Pressure placed gently against the side of a closed eye can sometimes produce a small flash of light. No light has entered the eye, but mechanical pressure has stimulated part of the visual system. Because the active pathway is associated with vision, the resulting experience is visual. In a similar way, activity within an auditory pathway is experienced as sound, while activity within particular bodily pathways may be experienced as touch, warmth, or pain. The nervous system interprets signals partly according to their route and destination. Not every stimulus is strong enough to be detected. The smallest amount of stimulation that can be noticed under specified conditions is called an absolute threshold. This is not a perfectly fixed border. Detection can vary with fatigue, attention, expectation, background noise, and many other factors. Another useful measure is the just noticeable difference. This is the smallest change between two stimuli that a person can reliably detect. The German physician Ernst Weber studied how noticeable differences depend on the starting intensity of a stimulus. Adding a small weight to an almost empty hand may be obvious, while adding the same amount to a heavily loaded hand may go unnoticed. The physicist and philosopher Gustav Fechner developed these ideas into psychophysics, the scientific study of relationships between physical stimulation and subjective experience. Psychophysics revealed that sensation does not usually increase in a simple one-to-one relationship with physical energy. The nervous system is sensitive to proportions, contrasts, and changes. This is one reason a candle is striking in a dark room, but barely noticeable in bright sunlight. The physical light from the flame may be similar, yet its perceptual effect depends strongly on the surrounding level of illumination. Sensory systems also adapt. When clothing is first placed against the skin, its pressure may be easy to notice. A short time later, the sensation often fades from awareness, even though the fabric remains in contact with the body. Entering a room with a noticeable smell may produce an immediate impression, only for that smell to become much less obvious after several minutes. Vision adapts as well. When moving from bright daylight into a dark room, very little may be visible at first. Over the following minutes, the visual system becomes increasingly sensitive. Rod photoreceptors gradually recover their ability to respond under low light, allowing shapes and surfaces to emerge from what initially seemed like darkness. Adaptation prevents the nervous system from spending equal attention on signals that remain unchanged. It makes changes, contrasts, and new events more prominent. A background hum, steady pressure, or persistent smell may gradually fade, while a sudden alteration becomes noticeable immediately. Long before conscious awareness, sensory systems are already compressing and filtering information. They compare neighbouring signals, emphasize boundaries, reduce repetition, and prioritize changes that may matter. The nervous system does not preserve every physical detail. It extracts patterns that are useful for recognition and action. Much of this information passes through the thalamus, a structure deep within the brain that acts as an important relay and processing centre. Visual, auditory, and many bodily signals travel through specialised regions of the thalamus before reaching areas of the cerebral cortex. Smell follows a somewhat different route. Olfactory information reaches the olfactory bulb and then travels to several cortical and limbic regions without first passing through the same kind of initial thalamic relay used by most other senses. Later stages of smell processing still interact with the thalamus and many other parts of the brain. It is useful then to separate sensation from perception. Sensation refers broadly to the detection and initial encoding of physical or chemical signals. Perception is the organization and interpretation of those signals into meaningful experiences. Sensation provides patterns of neural activity. Perception turns those patterns into a glowing lamp, a distant footstep, a cool breeze, or the faint smell of rain. By the time anything enters awareness, the nervous system has already begun deciding what matters, what has changed, and what the signals are most likely to mean. Vision often feels like the most direct of the senses. Open your eyes, and the world appears to be simply there, complete with colour, depth, movement, and solid objects. Yet the visual system does not receive a finished picture. It receives patterns of electromagnetic radiation and gradually transforms them into a useful model of the surroundings. Visible light is only a narrow portion of the electromagnetic spectrum. Human eyes are generally sensitive to wavelengths between roughly 400 and 700 nanometers, although the exact boundaries vary between individuals and depend on how sensitivity is measured. Shorter wavelengths extend into ultraviolet radiation, while longer wavelengths extend into infrared radiation. Both are physically present, but neither normally appears in human vision. When light reaches the eye, it first passes through the cornea, the clear curved surface at the front. The cornea provides much of the eye's focusing power. Light then travels through the pupil, the opening whose size is controlled by the coloured iris. Behind it lies the lens, which changes shape to help focus light from different distances. The focused light reaches the retina, a thin layer of nervous tissue lining the rear of the eye. Here, the process of vision truly begins. The retina contains two main classes of photoreceptor. Rod cells are highly sensitive and support vision under dim conditions, although they provide little colour information and relatively low spatial detail. Cone cells operate best in brighter light and support fine detail and colour discrimination. Cones are particularly concentrated near the centre of the retina, especially within a small region called the fovea, where visual acuity is greatest. Photoreceptors do not send information straight into the optic nerve. Their signals pass through an intricate network of retinal cells. Bipolar cells carry information between photoreceptors and retinal ganglion cells. Horizontal cells connect activity across neighbouring regions and help shape contrast. Amacrine cells influence timing, motion sensitivity, and communication among several retinal pathways. Ganglion cells gather the processed information, and their long fibres eventually form the optic nerve. The retina is therefore not passive photographic film. It is part of the brain, performing substantial processing before signals ever leave the eye. One important feature of retinal organization is the receptive field. A receptive field is the region of the visual scene that can influence the activity of a particular neuron. Many retinal ganglion cells have center-surround receptive fields. Light falling in the center may increase activity, while light in the surrounding region reduces it, or the arrangement may be reversed. This organization makes the visual system especially sensitive to contrast. A uniform patch of light often produces less activity than a sharp boundary between light and dark. Edges, changes, and local differences are emphasized because they are useful for identifying surfaces and objects. The visual system is less interested in recording every light level perfectly than in discovering where one thing ends and another begins. The fibres of the optic nerve carry this information towards the brain. At the optic chiasm, some fibres cross to the opposite side. This arrangement helps ensure that information from the left side of the visual world is processed mainly by the right cerebral hemisphere, while information from the right side is processed mainly by the left. Many visual signals then pass through the lateral geniculate nucleus of the thalamus. From there, they travel to the primary visual cortex in the occipital lobe at the back of the brain. Neuroscientists David Hubel and Torsten Wiesel studied how neurons in the visual cortex respond to specific patterns. They found cells that were especially sensitive to edges with particular orientations, as well as more complex patterns of movement and shape. Their work helped reveal that vision is built through layers of analysis, rather than received as one complete image. From the early visual cortex, processing continues along several interconnected routes. The ventral visual stream, extending towards areas of the temporal lobe, is associated largely with identifying objects, shapes, faces, and categories. It is sometimes described as the pathway concerned with what an object is. The dorsal visual stream extends towards the parietal lobe and is associated largely with spatial relationships, movement, and visually guided action. It helps estimate where objects are and how the body should move in relation to them. These streams interact extensively, so the division is useful but not absolute. Even the retina contains a region where no image can be detected. At the point where the optic nerve leaves the eye, there are no rods or cones. This creates the physiological blind spot. Yet people do not normally experience a dark hole floating in their vision. The brain uses information from the other eye and surrounding visual patterns to fill in the missing area without conscious effort. Vision also feels stable, despite the fact that the eyes are almost never still. They perform rapid movements called saccades several times each second, shifting the point of highest detail from one location to another. During these movements, visual sensitivity is briefly reduced through a process called saccadic suppression. Without it, the world might appear to smear whenever the eyes moved. Instead, separate glimpses are combined into a continuous and stable experience. What appears to be a direct view of reality is therefore the result of many hidden transformations. The visual system receives incomplete and constantly changing patterns of light. It enhances contrast, detects edges, separates motion, compares signals from both eyes, fills missing regions, and combines repeated glimpses across time. The result is not a perfect physical reconstruction of everything outside the body. It is a practical representation of surfaces, objects, distances, and boundaries, carefully shaped for recognition and action. The world seems visually effortless, only because the nervous system performs the difficult work quietly. Colour seems to belong naturally to the world. Grass appears green, the sky appears blue, and a ripe strawberry seems to carry its redness on its surface. Yet electromagnetic radiation does not arrive with colour already attached. It has wavelength, frequency, and energy. Colour is the experience created when the visual nervous system interprets particular patterns within that radiation. Human colour vision begins with cone photoreceptors in the retina. Most people have three broad classes of cone, commonly called short wavelength, medium wavelength, and long wavelength cones. These names describe the parts of the visible spectrum to which each class is most sensitive. They do not mean that one cone detects only blue, another only green, and another only red. Each class responds across a broad and overlapping range of wavelengths. A single wavelength can stimulate more than one cone type, but in different proportions. The brain compares those relative responses. This comparative system was central to trichromatic colour theory. The English physician and physicist Thomas Young proposed in the early 19th century that colour vision could be explained by a small number of receptor mechanisms rather than a separate receptor for every possible colour. The German scientist Hermann von Helmholtz later developed the idea further. Trichromatic theory explains how three cone classes can support the perception of an enormous range of colours. Different mixtures of light create different patterns of activity across the cone populations. The nervous system reads those patterns rather than measuring wavelength with a tiny internal ruler. Yet trichromatic theory alone could not explain every feature of colour experience. People do not normally perceive a colour that feels simultaneously reddish and greenish, or both bluish and yellowish in the same place. Certain colours seem to oppose one another. The German physiologist Ewald Hering developed opponent process theory to explain this organization. According to this view, the visual system compares color information along opposing channels. One channel contrasts red with green, another contrasts blue with yellow. A further channel compares light with dark. Modern neuroscience has shown that opponent processing emerges through several stages of the visual system, including retinal and later neural pathways. The trichromatic and opponent theories are therefore not competing descriptions in the simplest sense. They refer to different parts of the same process. Three broad cone classes first respond to light, and later circuits compare and reorganize their signals. This helps explain afterimages. Stare at a strongly colored object for a while and then look at a pale surface. A faint image may appear in an opposing color. A red object may leave a greenish afterimage, while a blue object may leave a yellowish one. The effect occurs because prolonged stimulation changes the responsiveness of parts of the color system. When the original stimulus disappears, the balance between opposing pathways is temporarily shifted. The perceived color depends not only on the new light entering the eye, but also on what the visual system has recently adapted to. Color is also shaped by neighboring regions. In simultaneous color contrast, the same central patch can appear different depending on the colors surrounding it. A grey square placed against a dark background may look lighter than an identical grey square placed against a bright background. The physical light from the square has not changed. Its perceptual appearance has changed because the visual system compares local signals. These comparisons are part of a broader ability called color constancy. The light reflected from an object can change dramatically throughout the day. A white page under bluish daylight reflects a different mixture of wavelengths than the same page under a warm indoor lamp. Yet the page usually continues to look white. The brain estimates the color of a surface by considering the likely illumination, the surrounding scene, and the relationships among nearby surfaces. It is not perfectly successful, as carefully designed photographs and unusual lighting can demonstrate, but in ordinary environments the process is remarkably effective. Without color constancy, familiar objects would seem to change color whenever a cloud crossed the sun or someone switched on a lamp. The visual system sacrifices exact measurement of reflected wavelengths in order to maintain a stable estimate of surface properties. Another important feature of color vision is metamerism. Two physically different mixtures of wavelengths can sometimes produce the same color experience. This happens when they stimulate the three cone classes in sufficiently similar proportions. A screen can create the experience of yellow by combining suitable amounts of red and green light, even though the mixture may contain no single wavelength corresponding to spectral yellow. The nervous system cannot distinguish every possible spectrum because it reduces the incoming light to the responses of three overlapping cone classes. Metamerism makes modern displays, color printing, and many lighting systems possible. It also provides a clear reminder that perceived color is not a complete record of the light spectrum. Not everyone has the same color vision. Inherited color vision deficiencies often involve differences in the genes that produce cone photopigments. The most common forms affect the photopigments associated with long wavelength or medium wavelength cones. This can make certain reds, greens, browns, and oranges more difficult to distinguish. The term color blindness can be misleading because many affected people still perceive a rich range of colors. Complete absence of color experience is much rarer than partial differences in color discrimination. Human color vision is only one arrangement among many found in nature. Many birds possess 4 types of cone photoreceptor and can detect ultraviolet wavelengths. Bees also respond to ultraviolet light and may see patterns on flowers that are invisible to humans. These patterns can help guide them towards nectar. Marine animals provide even more varied examples. Some fish have visual systems adapted to the filtered light of particular depths. Mantis shrimps possess an unusually large number of photoreceptor classes, although this does not necessarily mean they discriminate colour in the same detailed way that humans do. Their nervous systems may use a different strategy for identifying wavelengths rapidly. No species sees colour exactly as it exists, because colour is not a detachable substance waiting inside light. Each visual system samples part of the electromagnetic spectrum and transforms it according to its own receptors, neural circuits, environment, and evolutionary history. The red of a sunset, the green of a leaf, and the blue of deep water are therefore reliable biological interpretations. They correspond to real differences in light and real properties of surfaces, atmospheres, and illumination. But the experienced colours themselves are created through the activity of the visual system. The world does not arrive already painted. Light reaches the eyes, cone cells respond, neural pathways compare, and the brain quietly fills experience with colour. Each eye receives a pattern of light across a curved but essentially two-dimensional surface. Yet the world does not appear flat. It appears to contain distance, volume, movement, and solid objects that continue to exist as the eyes and body change position. This stable, three-dimensional experience is not present in either retinal image by itself. It must be constructed from many different clues. One of those clues is binocular disparity. Because the eyes are separated by several centimetres, each eye receives a slightly different view of nearby objects. The brain compares these differences and uses them to estimate depth. This contributes to stereoscopic vision, especially at relatively close distances. Hold one finger in front of your face and alternately close each eye. The finger appears to shift against the background because its position differs in the two retinal images. Normally, the visual system combines these views so smoothly that the shift is not noticed. Instead, it becomes information about distance. Binocular disparity is useful, but it is not essential for all depth perception. A painting, photograph, or cinema screen can look richly three-dimensional, despite presenting light on a flat surface. This is possible because the visual system also relies on monocular depth cues, which can operate through either eye alone. Linear perspective provides one familiar example. Parallel lines, such as railway tracks, appear to draw closer together as they recede into the distance. Relative size offers another clue. When two objects are believed to be similar in real size, the one producing the smaller retinal image is usually judged to be farther away. Texture gradients help transform a surface into depth. Stones, blades of grass, or marks on a road appear smaller and more densely packed as distance increases. Occlusion occurs when one object blocks part of another, indicating that the blocking object is closer. Shading can suggest curvature, hollows, and raised surfaces because the brain makes assumptions about how light usually falls across objects. Atmospheric haze also matters. Distant mountains often appear paler, less sharply defined, and slightly bluer because light is scattered as it travels through more atmosphere. The visual system can use this reduced contrast as a rough indication of distance. Movement supplies another powerful clue. During motion parallax, nearby objects appear to move rapidly across the field of view, while distant objects seem to move more slowly. Looking from the window of a moving train makes the effect obvious. A fence close to the track rushes past, while hills on the horizon appear almost still. The nervous system begins using some depth information early in life, although the development of depth perception is complex. In the 1960s, psychologists Eleanor Gibson and Richard Walk created the visual cliff experiment. A sheet of strong glass covered a patterned surface, with one side appearing shallow and the other appearing to drop away. Many crawling infants and young animals avoided crossing onto the apparently deep side, even though the glass made it physically safe. The result suggested sensitivity to visual depth. However, it does not prove that infants possess a fully mature adult understanding of height and danger. Behaviour may also depend on crawling experience. emotional signals from caregivers, species differences, and developing control of movement. Depth is only part of the problem. The visual system must also decide what is moving. Whenever the eyes turn, the entire image sweeps across the retinas. Similar changes occur when the head or body moves. Yet the room does not normally appear to slide whenever you glance towards the door. The brain combines retinal motion with information about eye movements, head movements, balance, and motor commands. This helps distinguish movement in the environment from movement produced by the observer. Even without real continuous movement, the brain can create the experience of motion from separate events. The psychologist Max Wertheimer studied the phi phenomenon during the early development of Gestalt psychology. When two stationary lights flash in rapid succession at different locations, an observer may perceive movement between them rather than two unrelated flashes. Cinema and animation rely on related forms of apparent motion. A rapid sequence of still images is experienced as continuous action. The screen does not contain a person physically moving through every intermediate position. The visual system supplies continuity by interpreting the changing sequence. Gestalt psychologists argue that perception is organized into meaningful wholes rather than built only from isolated sensations. Their principles describe some of the ways visual elements are grouped. Proximity encourages nearby features to be seen as belonging together. Similarity groups elements that share color, shape, orientation, or size. Good continuity encourages lines and contours to be perceived as following smooth paths. Closure allows incomplete boundaries to be experienced as complete shapes. Figure-ground organization separates a prominent object from the background against which it appears. These principles are tendencies rather than rigid laws. They reveal how the visual system searches for the simplest and most coherent organization available. Once an object has been identified, it usually appears stable despite dramatic changes in its retinal image. This is called object constancy. Size constancy allows a person walking away to remain perceptually similar in size, even though the image of that person shrinks across the retina. Shape constancy allows a circular plate to continue appearing round when viewed from an angle, even though its retinal image becomes more elliptical. The brain interprets these changing images in relation to estimated distance, viewpoint, lighting, and context. Without constancy, ordinary objects would seem to stretch, shrink, bend, and change shape every time either they or the observer moved. Motion perception presents further complications. When a moving edge is seen through a small opening, only the motion perpendicular to that edge may be directly visible. This ambiguity is known as the aperture problem. A diagonal line moving behind a narrow opening can appear to move in several possible directions because the local signal does not reveal the movement of the whole object. The brain resolves this by combining motion signals across larger regions. Areas of the visual cortex, including the middle temporal visual area, contain neurons strongly involved in processing direction and speed. By integrating many local measurements, the nervous system can estimate the movement of an entire surface or object. Yet these estimates can remain ambiguous. The Necker cube, for example, is a flat drawing that can be experienced as having two different three-dimensional orientations. The lines remain unchanged, but perception alternates between plausible interpretations. Visual camouflage exploits the same dependence on grouping and context. An animal may be physically present in plain sight while its colouring breaks up its outline, imitates surrounding textures, or makes figure-ground separation difficult. Once noticed, the animal can suddenly seem obvious, as though it has appeared from nowhere. In reality, the light reaching the eyes has barely changed. The organisation of the scene has. A stable world of separate objects is therefore an active perceptual achievement. The retinal images are incomplete, continuously moving and sensitive to lighting and viewpoint. From binocular differences, perspective, shading, texture, motion, context, and expectation, the brain constructs depth and continuity. The result is not a perfect copy of the shifting patterns projected onto the eyes. It is something more useful, a world of persistent objects that can be recognized, approached, avoided, picked up, and trusted to remain approximately where they were left. Sound begins with movement. A vibrating guitar string pushes and pulls the surrounding air. A loudspeaker moves rapidly backwards and forwards. Vocal folds disturb the air flowing through the throat. Thunder sends a powerful disturbance across the atmosphere. In each case, molecules in the air are compressed and spread apart in a travelling pattern of pressure variation. These pressure changes are mechanical waves. Unlike light, they cannot move through a perfect vacuum because they require matter to carry the disturbance. Sound can travel through gases, liquids, and solids, although its speed and behaviour depend on the material. In ordinary air near room temperature, it moves at roughly 340 metres each second. In water and many solids, it travels considerably faster. The frequency of a sound wave describes how many complete cycles occur each second. Frequency is closely related to pitch, although the relationship between physical frequency and experienced pitch is not perfectly simple. Faster cycles are usually heard as higher notes, while slower cycles are usually heard as lower ones. Amplitude describes the size of the pressure variation. Greater amplitude often contributes to greater perceived loudness, but loudness also depends on frequency, duration, background conditions, and the sensitivity of the listener. Waveform refers to the overall shape and mixture of frequencies within a sound. It contributes to timbre, the quality that allows the same musical note to sound different when played by a piano, violin, or human voice. The ordinary range of healthy young human hearing is often described as approximately 20 to 20,000 cycles per second. These boundaries are only rough guides. Sensitivity varies between people, differs across frequencies, and usually declines at the upper end with age. The highest sounds available to a child may be entirely absent from the audible world of an older adult, which is one of the quieter ways time announces itself. The journey into hearing begins with the outer ear. The visible structure on the side of the head collects sound and directs it into the ear canal. Its folds alter incoming waves in ways that later help the brain estimate whether a sound came from above, below, in front, or behind. At the end of the canal lies the eardrum, a thin membrane that vibrates in response to changing air pressure. These movements pass into the middle ear, where three tiny bones form a mechanical chain. They are called the malleus, incus, and stapes, also known as the hammer, anvil, and stirrup. The stapes is the smallest bone in the human body. Its movement presses against the oval window, a membrane leading into the fluid-filled inner ear. The middle ear bones help transfer sound energy from air into fluid. Without this arrangement, much of the vibration would be reflected rather than transmitted efficiently. Beyond the oval window lies the cochlea, a small spiral structure resembling a snail shell. Inside it, pressure waves move through fluid and produce travelling vibrations along the basilar membrane. The basilar membrane does not respond equally along its entire length. Regions near the base of the cochlea respond most strongly to higher frequencies, while regions farther towards the tip respond more strongly to lower frequencies. This ordered arrangement is called tonotopic organization. The Hungarian-American biophysicist Georg von Békésy investigated these travelling waves and showed how different frequencies reach their greatest vibration at different places along the cochlea. His work helped explain how the inner ear separates a complex sound into frequency-related components before the signals reach the brain. Resting upon the basilar membrane is the organ of Corti, which contains inner and outer hair cells. These cells are not covered with ordinary hairs. Their upper surfaces carry microscopic projections called stereotylia. As the basilar membrane moves, the stereotylia bend. This mechanical deflection opens tiny ion channels. Electrically charged particles enter the hair cells, altering their electrical state and leading to the release of neurotransmitters. Mechanical vibration has now become neural activity. Inner hair cells provide most of the sensory information sent towards the brain. Outer hair cells behave partly like biological amplifiers. They can change length in response to electrical activity, sharpening the movement of the basilar membrane and increasing sensitivity to quiet sounds. Fibers of the auditory nerve carry the resulting signals away from the cochlea. Information then passes through several structures in the brainstem, where timing, intensity, and differences between the two ears begin to be compared. From there, signals travel through the thalamus and onward to the auditory cortex within the temporal lobes. Hearing is not completed in a single brain region. Different networks contribute to locating sounds, recognizing voices, following speech, identifying musical patterns, and separating important signals from noise. To locate a sound, the nervous system compares information reaching the two ears. A sound arriving from one side usually reaches the nearer ear slightly earlier. This creates a binaural timing difference. The head also blocks part of the sound, especially at higher frequencies, so the nearer ear may receive a stronger signal. This creates a binaural intensity difference. The detailed shape of the outer ear adds further clues by altering different frequencies according to the sound's direction. The brain combines these timing, intensity, and spectral patterns to estimate where the source is located. It is an impressive calculation performed so quickly that it usually feels like no calculation at all. The physical wave reaching the ears is often a mixture produced by many sources at once. In a busy room, vibrations from voices, footsteps, ventilation, music, clinking glasses, and reflections from walls overlap in the same air. Yet these waves are not normally heard as one meaningless disturbance. The auditory system groups them into separate perceptual streams. The psychologist Albert Bregman described this process as auditory scene analysis. The brain uses clues such as common timing, similar pitch, shared rhythm, location, and gradual change to decide which parts of the signal probably came from the same source. A melody is grouped across time. One voice is separated from another. An echo is often attached to the sound that produced it rather than heard as an unrelated event. Context can even cause the auditory system to restore information that is physically missing. In phonemic restoration, part of a spoken word is replaced by a burst of noise, yet listeners may still hear the missing speech sound when the sentence makes it predictable. The brain uses language and context to supply the most likely interpretation. A related continuity illusion occurs when a tone is interrupted by a louder noise. If the timing and frequencies are suitable, the tone may seem to continue behind the noise even though it was briefly absent. The nervous system treats the interruption as an obstruction rather than as the end of the original sound. These effects do not mean hearing is careless. In natural environments, sounds are frequently masked by other events. Restoring a likely continuation allows the brain to follow voices and objects through noisy conditions. A melody, a spoken word, or a distant thunderclap is therefore not a sound object carried intact into consciousness. It begins as pressure changes moving through matter. The ear separates and transforms those changes, while the brain compares timing, frequency, intensity, location, memory, and expectation. From invisible vibration, the nervous system creates a world filled with voices, rhythm, distance, warning, music, and meaning. Vision and hearing can reveal events at a distance, but touch begins where the world meets the body. The skin marks a shifting boundary between what belongs to the body and what lies beyond it. Through touch, the nervous system detects contact, pressure, vibration, stretching, motion, and texture. It can recognize the weight of a hand resting on a table, the fine ridges of a coin, the softness of fabric, or the faint movement of air across the arm. Touch is not one single sense. It is a collection of specialized receptor systems distributed through the skin and deeper tissues. Different receptors respond best to different kinds of mechanical change, allowing the nervous system to build a detailed picture of what is happening at the body's surface. Merkel cells respond well to sustained pressure and fine spatial detail. They are especially useful for detecting edges, points, and the shape of small objects pressed against the skin. When a fingertip traces the raised lettering on a surface, Merkel cells contribute to the ability to identify those precise patterns. Meissner corpuscles respond strongly to light touch and movement across the skin. They are sensitive to relatively low-frequency vibration and help detect slipping. This allows the hand to adjust its grip before an object quietly escapes and falls to the floor, usually at the least convenient moment. Pacinian corpuscles are particularly sensitive to rapid vibration and sudden changes in pressure. Their layered structure allows them to respond strongly when stimulation begins or changes, while responding less to steady pressure. They help detect fine vibrations transmitted through tools and objects, including the subtle tremor felt through a pencil, a steering wheel, or the handle of a spoon. Roughney endings respond to sustained stretching of the skin. They contribute to information about finger position, hand shape, and the direction in which the skin is being pulled. These receptor categories are useful, although real touch perception depends on overlapping signals from several receptor types. The brain rarely receives a message meaning simply rough, smooth, hard, or soft. It receives patterns of activity that must be compared and interpreted. Sensitivity also varies greatly across the body. The fingertips and lips contain high densities of receptors with relatively small receptive fields. A receptive field is the area of skin whose stimulation can influence a particular sensory neuron. Smaller receptive fields allow nearby points to be represented separately. Larger receptive fields combine information across a wider region, providing less precise localization. This is one reason a tiny crumb on the lip may feel impossible to ignore, while a similar point of pressure on the back can be difficult to locate exactly. The two-point discrimination test provides a simple demonstration. Two nearby tips are pressed against the skin, and the person reports whether they feel 1 point or 2. On the fingertip, the points can often be distinguished when they are only a few millimeters apart. On the back or upper arm, they may need to be separated by several centimeters. These distances are not permanent measurements of an unchanging sensory map. Results depend on the method, pressure, attention, expectation, training, age, and individual variation. Even so, the test clearly reveals that spatial sensitivity is not distributed evenly. Signals from touch receptors travel along peripheral nerves towards the spinal cord. Much of the information involved in fine touch, vibration, and body position then ascends through pathways in the spinal cord to the brain stem. From there, it travels to the thalamus and onward to the primary somatosensory cortex in the parietal lobe. The pathways are organized according to location on the body. Neighboring regions of skin tend to be represented in neighboring regions of the nervous system, creating what is called a somatotopic map. The Canadian neurosurgeon Wilder Penfield explored this organization while operating on conscious patients during brain surgery. By applying gentle electrical stimulation to different areas of the cerebral cortex, Penfield and his colleagues could sometimes produce sensations that patients described as coming from particular body parts. This research contributed to the famous sensory homunculus, a distorted image of the human body arranged across the somatosensory cortex. The lips, tongue, hands, and fingers appear enormously enlarged, while the torso and limbs appear relatively small. The distortion reflects the amount of cortical territory associated with processing information from different regions, not the actual size of those body parts. It is also a simplified illustration rather than a perfectly precise map. The cortex contains overlapping, flexible representations that can change with learning, use, and injury. There is, regrettably, no tiny misshapen person stretched across the brain. The homunculus is only a diagram, which is probably for the best. Touch localization is sharpened through neural comparison. One mechanism involved is lateral inhibition. When one sensory pathway becomes strongly active, it can reduce activity in neighboring pathways. This increases contrast between the center of stimulation and the surrounding region. The result is similar to sharpening an edge in an image. a broad pattern of receptor activity can become a more precise estimate of where contact occurred. Lateral inhibition helps the nervous system distinguish nearby points and define the borders of an object pressing against the skin. Yet touch is most informative when the body moves. Active touch, also called haptic perception, involves deliberately exploring an object with the hands and fingers. Movement provides changing information about shape, texture, weight, hardness, temperature, and material. A coin hidden in a pocket can often be identified without sight. The fingers trace its circular edge, feel its thickness, detect the raised pattern on its surface, and compare its weight with familiar objects. No single receptor announces that the object is a coin. Recognition emerges from a sequence of movements, sensations, expectations, and memories. Texture depends especially on exploration. Coarse surfaces produce spatial patterns across the skin, while finer textures create rapid vibrations as the fingers move. The nervous system combines these signals with knowledge of hand movement to estimate what kind of surface is being touched. Judging whether fabric is rough, silky, thick, or worn involves much more than passive pressure. The fingers slide, squeeze, stretch, and release it. Expectations also matter. An object believed to be metal may feel colder than one believed to be wood, because materials conduct heat away from the skin at different rates, and prior knowledge helps interpret that change. Touch does more than describe external objects. It contributes to the feeling that the body has a boundary and belongs to the self. Contact on the skin is usually experienced as happening to me at a particular location on my body. This sense of ownership feels immediate, but it depends on the brain combining touch with vision, movement, body position, and prediction. When these signals agree, the body feels stable and unquestionably one's own. The skin therefore acts as both a sensory surface and a border of identity. Through countless patterns of pressure, movement, and vibration, the nervous system creates not only the feel of the world, but also the quiet impression of a body standing within it. Touch describes contact with the world, but the somatosensory system also creates experiences that feel more urgent. Warmth, cold, itch, irritation, and pain all help the body detect changing conditions and possible threats. They are protective sensations, although none of them provides a perfectly direct measurement of what is happening in the tissues. Temperature begins with specialized sensory neurons called thermoreceptors. These neurons respond when the skin becomes warmer or cooler. Their activity depends partly on proteins called transient receptor potential ion channels, which open or close under particular physical and chemical conditions. Research associated with David Julius helped reveal how some of these channels respond not only to temperature, but also to substances that create temperature-like sensations. Capsaicin, the active compound in chili peppers, activates a channel involved in detecting potentially harmful heat. Menthol activates a different channel associated with cooling. The molecules themselves are not literally heating or freezing the skin in the ordinary way. They are activating parts of the sensory machinery that normally respond to temperature. This is why mint can feel cool and chili can feel hot, even when both are close to room temperature. Ardem Pataputian and his colleagues identified important mechanically sensitive ion channels involved in touch and body awareness. Together, this work helped reveal how physical forces and temperature changes can be converted into electrical activity within sensory neurons. Temperature experience depends on more than the reading of a biological thermometer. Thermoreceptors respond strongly to change and then adapt. Water that initially feels warm may soon feel ordinary. A cool room can feel surprisingly comfortable after several minutes, while the same temperature may feel chilly when first entered. Comparison also matters. Place one hand in cool water and the other in warm water, then move both into water at the same moderate temperature. The water may feel warm to one hand and cool to the other. Its physical temperature is identical for both hands, but each sensory system is responding relative to its recent state. Some combinations of warm and cool stimulation can even create an experience of burning heat. This thermal grill illusion demonstrates that the nervous system integrates patterns across temperature and pain pathways, rather than assigning one simple sensation to each receptor. Itch is another distinct protective experience. It can be triggered by chemical irritation, inflammation, insect bites, dry skin, or immune activity. Histamine activates some itch-sensitive nerve fibers, although many forms of itch involve other pathways. An itch creates a powerful urge to scratch. Scratching can temporarily suppress the sensation by introducing mild pain and other competing signals, although it may also damage the skin and worsen irritation. The nervous system has designed a solution that is effective enough to be tempting and imperfect enough to keep dermatologists employed. Pain begins partly with nociceptors. These are sensory neurons that respond to potentially damaging mechanical pressure, extreme heat or cold, and certain chemical conditions. Nociceptors can warn of a sharp object, a burn, inflammation, or tissue strain. Nociception and pain are not the same thing. Nociception refers to the neural detection and transmission of potentially harmful conditions. Pain is the conscious unpleasant experience that may result from this activity. Pain is influenced by signals from the tissues, but it is also shaped by attention, expectation, emotion, memory, context, and activity descending from the brain. A painful sensation can become stronger when it is feared, watched closely, or interpreted as dangerous. It may become less intense when attention is elsewhere, or when the event is understood as controlled and temporary. This does not mean pain is imaginary. It means that pain is a biological judgment about threat and protection rather than a simple gauge displaying the exact amount of damage. The psychologists Ronald Melzack and Patrick Wall transformed scientific thinking about pain with their gate control theory. They propose that signals within the spinal cord can regulate how strongly nociceptive information continues towards the brain. Activity from touch and pressure pathways can sometimes reduce pain transmission, which helps explain why rubbing a bumped elbow may offer brief relief. Signals descending from the brain can also increase or decrease activity in these spinal pathways. The gate is not a literal little door opening and closing inside the spine. It is a useful description of interacting neural circuits that regulate sensory information before conscious pain emerges. Pain can also be felt far from its apparent source. In referred pain, signals from internal organs may be experienced as coming from the skin or muscles. Pain associated with the heart, for example, may be felt in the chest, arm, shoulder, neck, or jaw. One explanation is that sensory signals from internal organs and signals from regions of the body surface converge on some of the same neurons in the spinal cord. Because the brain receives far more experience interpreting signals from the skin than from internal organs, it may assign the source incorrectly. An even more striking example occurs after the loss of a limb. Many people continue to feel that the missing limb is present. It may seem to move, itch, tighten, or become painfully clenched. This is called a phantom limb experience. The limb is absent, but its representation within the nervous system does not immediately disappear. Brain regions, spinal pathways, peripheral nerve activity, memory, and the previous body model may all contribute. Neuroscientist Vilayana Ramachandran helped develop mirror box therapy for some forms of phantom limb pain. A mirror is positioned so that the reflection of the remaining limb appears to occupy the location of the missing one. Moving the intact limb can then create visual feedback suggesting that the phantom limb is also moving or relaxing. For some people, this altered feedback reduces painful sensations. Although results vary and mirror therapy is not a universal cure, its effects show how vision, movement, expectation, and body representation can influence pain. Expectation can also shape symptoms through placebo and nocebo effects. A placebo effect occurs when positive expectation and the treatment context contribute to measurable improvement. A nocebo effect occurs when negative expectation contributes to worsening symptoms or side effects. These effects can involve changes in attention, stress, learning, and the release of the body's own pain-regulating chemicals. They do not mean that symptoms are pretended, nor that a person can simply choose to stop feeling pain. Expectation is one influence within a much larger biological system. Pain can persist even after tissues have healed, especially when nervous pathways become sensitized. It can also be surprisingly mild during serious injury, particularly in moments of intense stress or danger. The relationship between damage and experience is therefore meaningful but not exact. Temperature, itch, and pain are constructed sensations, but constructed does not mean unnecessary or false. They usually guide the body towards protection, withdrawal, rest, cooling, warming, or attention. They are less like precise laboratory instruments and more like sensitive alarm systems. An alarm may respond to smoke before flames appear, remain active after danger has passed, or become overly sensitive. Even so, its purpose is clear. Through these sometimes uncomfortable experiences, the nervous system turns physical conditions into urgency, caution, and care. It does not merely report what has happened. It tries to keep the body safe. Close your eyes and lift one hand into the air. Even without seeing it, you probably know roughly where the hand is. You can tell whether the elbow is bent, whether the palm faces upwards, and whether the fingers are open or curled. This quiet awareness comes from proprioception, the sense of the position and configuration of the body. Proprioception is sometimes described as a hidden sense because it rarely attracts attention when it is working normally. Vision presents a visible world, hearing produces a world of sound, and touch becomes obvious whenever the skin meets an object. Proprioception works more quietly, providing a continuous estimate of where the limbs are and how they are arranged. Without it, even simple movement would become far more difficult. Reaching for a cup would require constant visual guidance. Walking would demand careful observation of every step. Scratching the back of the head might become a surprisingly ambitious project. Proprioceptive information comes from several kinds of receptor distributed through muscles, tendons, joints, and skin. Muscle spindles are specialized sensory structures found within skeletal muscles. They respond to changes in muscle length and to the speed at which a muscle is being stretched. When the forearm changes position, for example, muscle spindles provide information about the changing lengths of the muscles controlling the elbow. Golgi tendon organs are located where muscles connect with tendons. They respond to tension and force. Their signals help the nervous system estimate how strongly a muscle is pulling and contribute to the regulation of movement. Receptors around joints can provide information, particularly near the limits of joint movement. Stretching of the skin also contributes. As a finger bends, patterns of skin deformation change across the hand, offering additional clues about position. No single receptor announces the exact location of a limb. The brain combines activity from many sources with motor commands and previous sensory information. From these patterns, it maintains a working model of the body. The perception of bodily movement is often called kinesthesia. The terms kinesthesia and proprioception are sometimes used interchangeably, while other scientific and clinical descriptions use proprioception more broadly and reserve kinesthesia for the sense of movement itself. The terminology varies, but the underlying distinction is straightforward. The nervous system represents both where body parts are and how they are moving. Proprioception alone does not keep a person upright. Balance also depends heavily on the vestibular system within the inner ear. The vestibular organs sit beside the cochlea and contain fluid-filled structures that detect movements of the head. They include 3 semicircular canals, along with 2 chambers called the utricle and the saccule. The 3 semicircular canals are oriented in different planes, allowing them to respond to rotations in different directions. When the head begins to turn, fluid inside the canals lags slightly because of inertia. This bends a flexible structure containing sensory hair cells. The bending of microscopic projections on those hair cells changes the opening of mechanically sensitive ion channels. As in the auditory system, mechanical movement is converted into electrical and chemical signals. The semicircular canals respond mainly to rotational acceleration. They are especially informative when the head begins turning, stops turning, or changes rotational speed. During a steady rotation, the fluid gradually catches up, and the sensation of turning may weaken even though movement continues. The utricle and saccule are known as the otolith organs. They respond mainly to linear acceleration and to the direction of gravity. Their sensory surfaces contain tiny calcium carbonate crystals, called otoconia, resting on a gelatinous layer above the hair cells. When the head tilts or the body accelerates in a straight line, the weight and inertia of these crystals shift the layer and bend the hair cells. The resulting signals help indicate whether the head is upright, tilted, moving forwards, or moving vertically. The vestibular system also contributes to the stability of vision. When the head turns to one side, the eyes automatically move in the opposite direction. This response is called the vestibulo-ocular reflex. The reflex helps keep an image stable on the retinas while the head is moving. Without it, the visual scene would blur and bounce with every step. You can see the principle by holding a finger in front of you and turning your head from side to side while continuing to look at the fingertip. The eyes compensate almost immediately. Balance emerges from cooperation among vestibular signals, vision, proprioception, touch, and motor commands. The brain continually compares these sources and adjusts muscle activity to keep the body aligned. Standing on a firm floor in good light is relatively easy because the signals agree. Vision shows a stable room. Pressure beneath the feet indicates a level surface. Proprioception reports the positions of the ankles, knees, hips, and spine. The vestibular organs suggest that the head is upright. Close the eyes, and one source disappears. Most people can still stand, but the body often sways slightly more. Stand on an uneven or soft surface as well, and balance becomes harder because information from the feet and ankles is less reliable. Walking over stones or rough ground requires constant adjustment. The foot may land at an angle, the ankle may begin to tilt, and the body's centre of mass may shift. Touch and proprioception describe the surface and limb position. Vestibular signals indicate head movement, and vision identifies the next safe place to step. The nervous system blends all of this while movement continues. Problems can arise when normally compatible signals disagree. Motion sickness is one example. A person reading inside a moving vehicle may look at a page that appears visually stable. The eyes report very little movement, while the vestibular system detects acceleration, turning, and vibration. The mismatch can contribute to nausea, dizziness, sweating, and discomfort. A related conflict can occur in a stationary simulator or virtual reality environment. Vision may show rapid movement while the vestibular system reports that the body is sitting still. The precise causes of motion sickness remain an active area of research, but sensory conflict is one important explanation. After stepping onto land following a long boat journey, some people briefly feel as though the ground is still moving. The nervous system has adapted to repeated motion and must recalibrate when the environment becomes stable again. Even an ordinary lift can reveal the hidden work of these systems. As it begins moving upwards, the otolith organs respond to acceleration, and the body may briefly feel heavier. When the lift stops, the change can create a momentary impression of sinking. The physical movement is simple, but the experience depends on how acceleration and gravity are interpreted. The sense of being upright is therefore not a permanent reading from one internal instrument. It is a continually updated estimate. Signals from the inner ear, muscles, tendons, joints, skin, eyes, and motor system are compared many times each second. Small errors are corrected through changes in posture before they enter awareness. Most of the time, the result is a quiet impression of stability. that impression can feel so dependable that balance seems like a property of the world rather than an achievement of the nervous system. Yet darkness, uneven ground, illness, unusual acceleration, or conflicting visual information can quickly reveal how carefully it is maintained. To feel steadily located within space is to experience another construction of the brain. Gravity pulls, muscles tense, fluid shifts, eyes move, and receptors fire. From all of it, the nervous system creates the simple and reassuring feeling that the body knows which way is up. Smell and taste allow the nervous system to explore the chemical world. They help identify food, detect smoke or decay, recognize familiar places, and monitor conditions inside the mouth and body. Although they are often treated as smaller companions to vision and hearing, chemical senses shape much of what feels comforting, appetizing, unpleasant, dangerous, or deeply familiar. Smell begins when volatile molecules travel through the air and enter the nasal cavity. Near its upper surface lies the olfactory epithelium, a specialized patch of tissue containing millions of olfactory receptor neurons. Airborne molecules dissolve into the thin layer of mucus covering this tissue. They can then bind to receptor proteins extending from the sensory neurons. Binding alters activity within the cell and begins the conversion of chemical information into electrical signals. Humans possess hundreds of functional olfactory receptor types. Each receptor can respond to several molecular structures, and each odor molecule may activate more than one receptor. A smell is therefore represented by a pattern of activity across many receptor populations rather than by one receptor devoted exclusively to coffee, roses, or wet pavement. The American scientists Linda Buck and Richard Axel helped reveal the large family of genes that encode olfactory receptors. Their discoveries showed how a limited collection of receptor types could support discrimination among an enormous variety of odors through combinatorial coding. Fibers from olfactory receptor neurons pass through tiny openings in the skull and enter the olfactory bulb. Here, neurons carrying the same receptor type tend to converge within structures called glomeruli. Different molecules create distinctive patterns across these glomeruli, giving the brain an organized representation of the chemical mixture entering the nose. Information then travels towards several brain regions. These include the piriform cortex, which contributes to identifying and representing odors, as well as the amygdala, entorhinal cortex, and orbitofrontal regions. These connections help link smell with emotion, memory, reward, decision-making, and flavour. Smell is often said to bypass the thalamus, but this statement requires care. Initial olfactory signals can reach primary olfactory cortical regions without first passing through the type of thalamic relay used by vision, hearing, and much bodily sensation. Later, olfactory processing does involve the thalamus, particularly in pathways connected with attention, identification, and conscious evaluation. The close relationship between olfactory regions, the amygdala, and memory-related structures helps explain why a smell can sometimes evoke an unusually vivid autobiographical memory. A particular soap, perfume, meal, or damp woodland may suddenly restore the atmosphere of a place not visited for many years. The smell does not contain the memory. Instead, neural activity triggered by the odour activates associations formed during earlier experience. Because smells are often encountered alongside emotionally meaningful events, those associations can become especially powerful. Taste begins when substances dissolve in saliva and interact with taste receptor cells. These cells are grouped within taste buds found mainly on structures called papillae across the tongue. But taste buds also occur in other parts of the mouth and throat. The familiar tongue map, with sweetness restricted to the tip and bitterness confined to the rear, is inaccurate. Different regions may show modest variations in sensitivity, but the major taste qualities can generally be detected across broad areas containing suitable receptors. The 5 widely recognized basic taste qualities are sweet, sour, salty, bitter, and umami. Sweetness commonly indicates sugars and other energy-rich substances. Saltiness responds largely to electrically charged sodium particles. Sourness is associated with acidity. Bitterness can signal many different compounds, including some that are potentially harmful, although plenty of perfectly safe foods are bitter as well. Umami is a savory taste associated especially with glutamate and certain related compounds. The Japanese chemist Kikunae Ikeda investigated the distinctive taste of seaweed broth and identified glutamate as its chemical basis. He named the sensation umami, meaning broadly delicious savouriness. Researchers continue to investigate whether other oral sensations, including responses to fat, calcium, water, or particular carbohydrates, should be considered additional basic tastes. Some evidence supports specialized mechanisms, but scientific classification remains more complicated than simply adding every recognizable food quality to a growing list. Taste alone provides only a small part of what is commonly called flavour. Much of flavour depends on smell, especially retronasal olfaction. During chewing and swallowing, volatile molecules are released from food and travel from the mouth up into the nasal cavity. There, they stimulate olfactory receptors from behind. The resulting experience seems to come from the food inside the mouth rather than from the nose. This is why flavour can become strangely flat during a cold or when the nose is blocked. Sweetness, saltiness, sourness, bitterness, and umami may still be detectable, but the detailed identity of coffee, fruit, herbs, chocolate, or roasted food becomes much harder to recognize. Flavor also includes texture, temperature, and sound. Crispness depends partly on vibrations and noises produced during biting. Creaminess involves patterns of touch and movement across the mouth. Warmth changes aroma release and can alter sweetness or bitterness. Vision contributes expectations about ripeness, freshness, and intensity before the food even reaches the tongue. Another part of chemical experience is chemesthesis. This refers to sensations produced when chemicals activate pathways associated with touch, temperature, irritation, or pain, particularly through branches of the trigeminal nerve. Menthol activates temperature-sensitive channels and creates a cooling sensation without necessarily lowering tissue temperature very much. Capsaicin activates channels associated with potentially harmful heat, creating the familiar burn of chili. Carbonated drinks produce tingling and mild irritation, while compounds in mustard, wasabi, and onions stimulate other chemical warning pathways. These sensations are not ordinary tastes, even though they contribute strongly to eating and drinking. A chili does not taste hot in the same way that sugar tastes sweet. It convinces part of the nervous system that something thermally alarming is happening. Despite arriving at the table at a perfectly reasonable temperature, expectation can reshape flavour as well. Food colouring may cause a drink to be identified as a flavour associated with its colour, even when the chemical mixture suggests something else. A pale drink coloured red may seem fruitier, while identical foods described differently can receive different ratings of sweetness, richness, or freshness. None of this means flavour is imaginary. Real molecules stimulate real receptors and produce measurable neural activity. Yet the final experience is assembled from many sources. Taste, smell, touch, temperature, irritation, vision, memory, and expectation all contribute. Food therefore does not deliver flavour intact, like a parcel placed directly into consciousness. It releases chemicals and creates physical changes. The nervous system organises those signals into the warm complexity of soup, the sharpness of citrus, the comfort of familiar bread, or the regrettably lasting enthusiasm of an especially powerful chili. The senses may begin in different organs, but conscious experience does not usually arrive as a collection of separate channels. A barking dog is not presented as one visual event, one sound, one patch of fur, and one moving shape that must be consciously assembled. It appears as a single animal occupying one place, producing one voice, and interacting with the surrounding world. This unification is called multisensory integration. The brain combines information from vision, hearing, touch, smell, taste, balance, and body position to estimate what is happening, where it is happening, and which signals probably share a common cause. The result is a coherent scene containing objects that seem able to look, sound, feel, smell, and taste a particular way. This process matters because the senses do not provide identical kinds of information. Vision is usually precise about spatial layout, but can be relatively slow to reveal sudden timing. Hearing is highly sensitive to rapid changes in timing, but may locate a source less precisely. Touch provides direct information about contact. Smell and taste reveal chemicals. The vestibular system contributes movement and orientation. By combining them, the nervous system can produce a more reliable estimate than any one sense could provide alone. An important structure involved in linking sensory information with rapid behavior is the superior colliculus, located in the midbrain. It contains organized maps related to visual, auditory, and bodily space. Activity there helps direct the eyes, head, and attention towards significant events. A sudden movement at the edge of vision and a sharp sound from the same direction may jointly trigger a faster and stronger orienting response than either signal alone. The superior colliculus contributes to the almost automatic turn towards the source, often before a person has clearly identified what happened. Neuroscientists Barry Stein and Alex Meredith conducted influential research on multisensory integration, particularly within the superior colliculus. Their work showed how neurons can respond to information from more than one sense, and how combined signals may enhance or alter neural activity. The brain is especially likely to combine signals when they correspond in time and space. Temporal correspondence means that events occurring at roughly the same moment are more likely to be interpreted as belonging together. Spatial correspondence means that signals appearing to originate from the same location are more likely to be assigned to one cause. A flash and a bang occurring together in the same direction are likely to be experienced as one event. If the sound arrives much later, or from somewhere else, the brain becomes less certain that the two are connected. This process is not perfectly strict. Sound travels more slowly than light, so a distant explosion is seen before it is heard. The nervous system can tolerate some delay when the physical situation makes that delay plausible. Vision often has a strong influence over perceived location. The ventriloquism effect provides a familiar example. A performer speaks without visibly moving the lips while a nearby puppet appears to talk. The sound reaches the ears from the performer, but the moving mouth of the puppet offers a convincing visual source. The apparent location of the voice is pulled towards the puppet. The auditory signal has not physically moved. The brain has assigned it to the visual event most likely to have caused it. The same principle operates when watching television or cinema. A voice seems to come from the actor's mouth, even though the sound may actually be produced by speakers positioned elsewhere in the room. Vision can also alter what speech sound is heard. In the McGurk effect, described by psychologists Harry McGurk and John MacDonald, a person hears one speech sound while watching a face produce a different mouth movement. The resulting experience may be a third sound that was present in neither the visual nor the auditory signal alone. For example, hearing a sound resembling bay while watching lips form, gay may produce the perception of day. The precise result varies between people and conditions, but the effect demonstrates that speech perception is audio-visual. The brain does not simply listen to speech, it also watches it. The influence can work in the opposite direction. Hearing can change what appears to be seen. In the sound-induced flash illusion, A single visual flash accompanied by two brief sounds may be perceived as two flashes. The auditory system provides precise timing information, and that information can alter the organization of the visual event. The person is not pretending to see an extra flash. The integrated percept genuinely differs from the physical visual stimulus. Flavour offers an even broader example. What is casually described as the taste of food is assembled from several sensory systems. Taste receptors contribute sweetness, sourness, saltiness, bitterness, and umami. Retronasal smell supplies much of the food's detailed identity. Touch reveals creaminess, crispness, thickness, and dryness. Temperature affects both sensation and the release of volatile molecules. The trigeminal system contributes the burn of chili, the cooling of menthol, and the fizz of carbonation. Vision shapes expectation through colour and appearance. Sound contributes the crack of a crisp or the crunch of fresh vegetables. These signals are experienced as one flavour belonging to one piece of food. The brain does not ordinarily label each contribution separately. It combines them into a unified estimate of what is being eaten. Researchers often describe multisensory perception using Bayesian models. These mathematical approaches propose that the nervous system combines current sensory evidence with prior knowledge about what is likely to occur. Signals are also weighted according to their estimated reliability. A clear visual signal may dominate a vague sound when judging location. In darkness, hearing may become more influential because the visual evidence is poor. Scientists Mark Ernst and Martin Banks studied this kind of reliability-based integration using maximum likelihood models. In their experiments, people combined visual and touch information in ways that closely resembled an optimal statistical estimate. The more reliable sense received greater weight, helping reduce overall uncertainty. This does not require a person to perform conscious mathematics. The framework describes the pattern of behaviour, not an inner mathematician sitting behind the forehead with a small calculator. Sensory dominance is therefore context-dependent. Vision often dominates spatial judgments because it usually provides detailed information about position and shape. Hearing frequently dominates fine timing because the auditory system is especially sensitive to rapid changes. Touch becomes decisive when an object is directly handled. Smell can strongly influence flavour when the nose is clear, but contributes far less when congestion blocks retronasal airflow. The balance shifts according to the quality of the available evidence. Most of the time, the senses agree well enough that their separate origins remain hidden. A cup appears to be one object. Its outline is seen, its weight is felt, its contact with the table is heard, and its contents may be smelled and tasted. Yet all of these signals arrive through different receptors, travel at different speeds, and follow different neural pathways. The brain must decide which belong together, which should dominate, and whether their timing and location support a common cause. The seamless world of experience is therefore not delivered by any single sense. It is assembled through constant negotiation among them. Vision, hearing, touch, smell, taste, balance, and bodily awareness contribute partial information. From those separate signals, the nervous system creates one event, one object, one place, and one continuous reality. At every moment, the nervous system receives far more information than conscious awareness can hold at once. Light reaches wide areas of the retinas. Sounds arrive from several directions. Clothing presses against the skin. Muscles signal their position. The inner ear reports movement and gravity. Smells drift through the air, while the body quietly supplies information about breathing, temperature, hunger, and tension. Most of this does not become the clear focus of experience. Instead, attention selects certain signals for deeper processing, while others remain faint, are handled automatically, or fail to enter awareness altogether. This process is known as selective attention. Selective attention does not completely shut down the rest of the sensory world. Background information may still influence behaviour, guide movement, or suddenly become important. But only a small part of the available scene can usually be examined in detail at one time. A busy social gathering offers a familiar example. Several people may be speaking nearby, music may be playing, glasses may be clinking, and chairs may be moving across the floor. Yet a listener can usually follow one conversation while much of the surrounding noise fades into the background. The British psychologist Colin Cherry investigated this ability during the 20th century. His work helped establish what became known as the cocktail party problem. Cherry presented different spoken messages to the two ears and asked listeners to repeat one while ignoring the other. People could follow the selected message reasonably well, but they usually remembered very little of the unattended content. They could often notice basic changes, such as a switch from a male to a female voice, or from speech to a tone, without identifying the meaning of the ignored words. This suggested that attention can favour one stream while reducing the processing of competing information. The psychologist Donald Broadbent proposed an early filter model of attention. According to this account, sensory information is briefly available, but a filter selects one stream for further processing on the basis of physical features such as location or voice. Unselected information is largely blocked before its meaning is fully analysed. The model captured the limited capacity of attention, but later findings suggested that ignored information is not always excluded completely. People may notice their own name spoken in an unattended conversation, for example, even though they cannot report most of what was said. Anne Treisman developed attenuation theory to explain this. Rather than treating attention as a gate that closes entirely, she suggested that unattended signals are weakened. Important or highly familiar information may still reach awareness, because it requires less activation to be recognized. Attention, in this view, behaves more like a volume control than a sealed door. The limits of awareness become especially striking in inattentional blindness. This occurs when a clearly visible event goes unnoticed because attention is directed elsewhere. Psychologists Arian Mack and Irvin Rock carried out influential experiments showing that observers could fail to notice unexpected objects appearing near the center of vision while they concentrated on another task. Daniel Simons and Christopher Chabry later demonstrated the effect in a widely known study involving people passing basketballs. Observers were asked to count passes made by one team. During the scene, an unexpected person in a gorilla costume walked through the group. A substantial proportion of viewers failed to notice the gorilla because their attention was occupied by the counting task. The object was not hidden. It crossed a visible part of the scene and remained present for several seconds. Yet visibility alone was not enough to guarantee awareness. This does not mean that the eyes failed to receive light from the figure. The visual system registered at least some information from that region. What failed was the deeper selection and interpretation required for the event to become part of conscious experience. Change blindness reveals a related limitation. Large alterations in a scene can go unnoticed when the visual interruption accompanying the change prevents attention from being directed to the correct location. In laboratory demonstrations, one image may alternate with a slightly altered version, while a brief blank screen appears between them. A building can disappear, an object can change colour, or a person's clothing can be replaced, yet observers may take surprisingly long to identify the difference. Similar effects occur outside the laboratory. During a conversation, one person can sometimes be replaced by another, while a large object briefly passes between them. Some participants continue speaking without immediately noticing the substitution. The nervous system does not maintain a perfectly detailed inner photograph of every feature in sight. It stores selected information, general structure, and features that appear important. When attention is disrupted, even substantial changes may fail to receive the comparison needed for detection. Looking directly at something does not guarantee conscious perception either. Eye fixation and attention are related, but they are not the same process. A person may stare towards a page while thinking about something entirely different and absorb almost none of the text. Conversely, attention can shift towards something in peripheral vision before the eyes move to inspect it. The point of gaze identifies where the eyes are directed, while attention determines which information receives priority. Some attention is guided from the top down. Top-down attention is shaped by goals, expectations, knowledge, and current tasks. Searching for a friend in a crowd makes familiar faces more important. Looking for a red suitcase increases sensitivity to red shapes. A musician listening for the bass line can bring one part of a familiar recording into the foreground. Other shifts are driven from the bottom up. Bottom-up attention is captured by features that are sudden, intense, contrasting, novel, or potentially important. A loud crash, a bright flash, unexpected movement, or a voice calling from behind can interrupt an existing goal. These systems often cooperate, but they can also compete. A reader may intend to concentrate on a quiet paragraph, while a nearby notification makes an energetic attempt to renegotiate that arrangement. Attention also has limits across time. The attentional blink occurs when people view a rapid sequence of items and try to identify two targets. If the second target appears shortly after the first, it is more likely to be missed. Processing the first target seems to occupy limited attentional resources for a brief period. The second item may be detected at an early sensory level, yet fail to receive enough processing to become reportable awareness. The blink usually lasts only a fraction of a second, but it reveals that conscious selection cannot always be repeated instantly. Attention, therefore, does more than brighten one part of an already complete internal picture. It helps determine which features are analysed deeply, connected with meaning, stored in memory, and admitted into the experienced world. Two people can occupy the same physical environment while noticing very different things. A witness may remember a face but not the colour of a vehicle. A driver may monitor movement on the road while overlooking a familiar building. A proofreader notices misplaced words that an ordinary reader passes over. A musician hears a timing error hidden inside an otherwise convincing performance. Experts often perceive more meaningful structure not because their eyes or ears are fundamentally stronger, but because experience has trained attention towards useful patterns. A radiologist learns where to look within a medical image. A birdwatcher notices a shape or call that others treat as background. A chess player sees strategic arrangements where a beginner sees individual pieces. The external information may be shared, but the selected reality is not identical. Attention allows the nervous system to operate within a world too rich to process evenly. It chooses, suppresses, shifts, and occasionally misses something wearing a gorilla costume. Through that selection, only part of the available world becomes the world that is consciously noticed. Perception depends on signals arriving from the world, but those signals are rarely complete. Light may be dim, speech may be partly masked, and objects may be hidden behind one another. Even under good conditions, sensory information can be noisy and ambiguous. The nervous system solves this problem partly by using what it already knows. Past experience creates expectations about which objects, sounds, movements, and events are likely to occur. These expectations help the brain interpret uncertain sensory evidence quickly. Perception is therefore influenced from two directions at once. Information travels inward from sensory receptors, while knowledge and expectations influence how that information is organized. The German physician and physicist Hermann von Helmholtz described perception as a form of unconscious inference during the 19th century. Helmholtz argued that the nervous system draws conclusions about the causes of sensory signals without requiring deliberate reasoning. When a flat pattern of light reaches the retina, the brain does not merely register that pattern. It infers that the pattern was probably caused by a particular object at a particular distance under particular lighting conditions. The reasoning is unconscious because the person experiences only the result. A chair simply appears to be a chair. The hidden process that produced that conclusion remains unnoticed. Modern predictive processing develops this general idea into a broader framework. Researchers including Carl Friston, Andy Clark, Anil Seth, and Jacob Howe have argued that prediction may be fundamental to perception, and perhaps to many other brain functions. Predictive processing is not one single theory accepted in exactly the same form by every scientist. It is a family of related models. They share the general proposal that the brain continually uses internal models to predict incoming sensory signals and then updates those models when the predictions are inaccurate. A top-down prediction travels from higher levels of processing towards lower sensory levels. It represents what the system expects to encounter. Bottom-up information arrives from receptors and early sensory pathways. When the incoming signal differs from the prediction, the difference is often described as a prediction error. The nervous system may then adjust its interpretation, alter the importance assigned to the signal, or update its future expectations. Prediction in this context does not mean consciously guessing what is about to happen. A person does not need to sit quietly and decide that the next object will probably be a table. The process is automatic. It depends on regularities learned across years of experience. Reading provides a simple example. Written language contains predictable patterns. Familiar words can often be recognized even when a letter is partly hidden or blurred. The surrounding sentence narrows the possibilities, allowing the brain to interpret an imperfect visual signal. The same thing happens during speech perception. A sentence heard in a noisy room may contain gaps where several sounds are masked. Knowledge of language, grammar, context, and the speaker's topic helps restore the most likely words. The listener experiences a continuous sentence rather than a collection of uncertain fragments. Familiar faces also depend on prediction. A face seen from an unusual angle, partly covered by hair, or briefly illuminated, may still be recognized because the brain has learned which features tend to occur together. The available evidence is matched against stored knowledge of the person. Partially hidden objects are interpreted in much the same way. A cat disappearing behind a sofa is not usually experienced as two unrelated pieces of animal, one visible at each side. The brain assumes that a complete body continues behind the obstruction, because this is the most probable cause of the visible fragments. In low light, prediction becomes especially important. A vague shape near the door may initially appear to be a person, because that interpretation fits familiar possibilities. More sensory evidence may reveal that it is only a hanging coat. The first perception was not random. It was a plausible conclusion formed from limited information, even if the conclusion required correction. The hollow mask illusion demonstrates how strongly prior expectations can influence visual experience. When the inside of a hollow mask is viewed from a suitable distance, it may appear to project outward like an ordinary face. The sensory evidence contains clues indicating a concave surface. Yet human observers have extremely strong expectations that faces are convex. The brain favours the familiar interpretation, sometimes causing the mask to appear to turn in an unusual direction as the observer moves. This illusion reveals that perception is not determined by sensory input alone. Expectations about the typical shape of faces can temporarily outweigh some of the available depth information. Predictive models are often connected with Bayesian inference. In Bayesian terms, the brain combines current evidence with prior probabilities, usually called priors. A prior represents how likely a possible explanation seemed before the latest sensory signal arrived. Seeing a small moving shape in a city park may create several possibilities. It might be a bird, a leaf, or a piece of litter. Previous experience assigns different probabilities to those explanations. The new sensory evidence then modifies them. A fluttering movement may support the bird's interpretation. A steady path along the ground may favour wind-blown litter. Perception settles on the explanation that best combines what is currently detected with what has usually proved likely in similar situations. Experience continually reshapes these priors. A doctor becomes more likely to recognise meaningful patterns in a medical image. A musician becomes more sensitive to the expected structure of harmony and rhythm. Someone who has repeatedly encountered a dangerous animal may interpret ambiguous movement differently from someone without that history. Not all sensory signals should receive equal influence. Predictive processing models often include the idea of precision weighting. Precision refers broadly to the estimated reliability of a signal or prediction. Clear sensory evidence is given more weight than uncertain evidence. A sharply visible object in daylight can strongly correct a mistaken expectation. A vague shadow at night leaves greater room for prior knowledge to guide the result. Attention may partly operate by changing precision. A signal judged important becomes more influential, while irrelevant or unreliable information contributes less. This balance helps prevent perception from becoming either completely rigid or completely unstable. If predictions always dominated, the brain would ignore evidence and remain trapped in expectation. If every sensory fluctuation received equal weight, perception would become overwhelmed by noise and constant minor variation. Prediction also improves speed and efficiency. The world contains many repeated patterns. Walls remain still, familiar rooms retain their general layout, and ordinary sounds unfold in recognizable ways. Reconstructing every detail from the beginning at every moment would require enormous effort. Instead, the nervous system can predict the continuing pattern and devote more processing to unexpected changes. A steady background sound becomes less noticeable, while a sudden interruption attracts attention. A familiar face is recognized quickly, while an unusual expression receives further analysis. Unexpected information is especially valuable because it may signal danger, opportunity, or the need to update an old belief. Prediction does not mean that human beings invent reality freely. Sensory evidence places strong constraints on what can be perceived. Expectations may influence an ambiguous shape, but they cannot usually transform a clearly visible tree into an ocean, or make a silent room produce an ordinary conversation. The brain's model must remain accurate enough to support successful action. Predictions that repeatedly fail are adjusted. Physical reality keeps correcting the nervous system, sometimes rather firmly. Scientists continue to debate how far predictive processing should be taken. Some researchers view it as a broad unifying account of perception, movement, attention, and bodily regulation. Others regard it as a useful family of mathematical models that applies well to some processes but not necessarily to all brain activity. Critics also warn that the framework can become too flexible. If almost any result can be explained afterwards as a prediction error, or change in precision, the theory risks becoming difficult to test. A valuable scientific account must make clear predictions of its own and identify evidence that could prove it wrong. Even with these debates, the central insight remains powerful. Perception is not a one-way flow from world to mind. Incoming signals interact with knowledge accumulated across a lifetime. Experienced reality emerges through this controlled meeting between evidence and expectation. The senses provide constraints, changes, and fragments. The brain supplies learned structure, likely causes, and possible meanings. What appears in consciousness is neither a raw copy of the external world nor a private invention detached from it. It is the nervous system's best current interpretation of what the world is most likely to contain. Perceptual illusions are often treated as entertaining failures of the senses. A line looks longer than an identical line. A still image appears to move. A grey square looks bright in one setting and dark in another. These effects can be amusing, but their scientific value reaches far beyond the momentary surprise. Illusions reveal the rules that perception normally follows. The nervous system must interpret incomplete and uncertain sensory information quickly. To do this, it relies on comparison, context, adaptation, grouping, learned expectations, and assumptions about the likely structure of the world. In ordinary settings, these methods are extremely effective. An illusion is often created by placing those useful methods in an unusual situation, where the resulting perception differs from a physical measurement. There is no single perfect classification of illusions, because many examples involve several mechanisms at once. Even so, a few broad categories are helpful. Physical illusions begin partly with the behaviour of light, sound, or another signal before it reaches the sensory receptors. A straw appears bent in water because light changes direction as it passes between air and water. A mirage can occur when layers of air at different temperatures bend light along curved paths. Physiological illusions arise more directly from adaptation or interactions within sensory pathways. After images and motion after effects belong broadly to this group. Ambiguous figures occur when one unchanged stimulus supports more than one reasonable interpretation. Cognitive or context-dependent illusions are strongly influenced by surrounding patterns, assumptions, depth cues, or learned expectations. These categories overlap because perception is not divided into neat little boxes merely to keep textbooks tidy. The Mueller-Lyer illusion consists of lines with arrow-like shapes attached to their ends. One line has fins pointing inward, while another has fins pointing outward. Although the central lines may be physically equal, one usually appears longer. Several explanations have been proposed. The surrounding angles may influence the estimated positions of the endpoints. Learned associations with depth and corners may also contribute under some conditions. No single explanation accounts perfectly for every variation, but the illusion clearly shows that length is judged in relation to context rather than measured in isolation. The Ponzo illusion uses converging lines resembling railway tracks receding into the distance. Two horizontal bars of equal length are placed at different positions between them. The higher bar, which appears farther away, often looks longer. The visual system interprets the converging lines as a depth cue. If two objects create similar retinal images but one appears more distant, the distant object would normally need to be physically larger. Size constancy may therefore influence the judgment, enlarging the apparent size of the bar that seems farther away. In the Ebbinghaus illusion, a central circle is surrounded by either larger or smaller circles. The central circle surrounded by large shapes usually appears smaller than an identical circle surrounded by small shapes. Apparent size is altered through comparison with neighbouring objects. Together, these illusions show that the nervous system does not carry an internal ruler that measures every object independently. It estimates size and length from relationships within the scene. Brightness and lightness are similarly dependent on context. Brightness refers broadly to the perceived intensity of light, while lightness concerns the estimated reflectance of a surface, meaning how light or dark that surface appears to be as a material property. Vision scientist Edward Adelson demonstrated this dramatically with the checker shadow illusion. A checkerboard contains a shadow cast across part of its surface. Two squares that reflect the same amount of light can appear very different, with one seeming much lighter than the other. The brain does not merely record the light arriving from each square, it tries to separate the properties of the surface from the effects of illumination. A square inside shadow is interpreted as receiving less light, so the visual system compensates and estimates that its surface must be lighter. This correction is usually useful. It allows a white object to remain recognizably white in shade, and a dark object to remain dark in bright sunlight. In Adelson's carefully arranged image, however, the correction produces a powerful mismatch between appearance and measurement. Adaptation can also make still scenes seem to move. Stare at a waterfall for some time and then look at the nearby rocks. The rocks may appear to drift upwards, even though they remain still. This motion after effect is often called the waterfall illusion. Aristotle described a related experience in ancient Greece, and later researchers examined it experimentally. Neurons sensitive to downward movement adapt during prolonged viewing. When the motion stops, the balance among motion-sensitive populations is temporarily altered, producing the impression of movement in the opposite direction. Ambiguous figures expose a different feature of perception. The Necker cube is a simple line drawing of a transparent cube. Because the image contains limited depth information, either of two faces can appear to be at the front. Perception may alternate between the two arrangements even though the drawing does not change. The Rubin vase produces a similar reversal in figure-ground organization. One moment, a central white vase appears against a dark background. The next, The dark regions become two faces looking towards one another, while the white region becomes background. Both interpretations are supported by the same sensory input. What changes is the organisation imposed upon it. Binocular rivalry provides an even more striking demonstration. Different and incompatible images are presented separately to the two eyes. Instead of blending smoothly into one stable picture, conscious awareness may alternate between them. One image dominates briefly, then the other appears, sometimes with fragments of each moving through awareness. The signals reaching the eyes remain relatively constant, yet conscious perception changes. This suggests that awareness depends not only on sensory stimulation, but also on competition and selection among neural representations. The British psychologist Richard Gregory described perception as a process of hypothesis testing. According to his approach, the brain uses sensory evidence to evaluate possible explanations of what caused the input. Illusions occur when the most plausible perceptual hypothesis under ordinary conditions happens to be wrong in an artificial or unusual display. This view fits well with many examples, although illusion science includes several competing and complementary explanations. Some effects can be understood through early neural interactions. Others depend more heavily on depth, experience, attention, prediction, or statistical properties of natural scenes. An illusion often remains compelling even after its construction has been explained. Knowing that two lines are equal does not necessarily make them look equal. Understanding the checker-shadow illusion does not immediately force the squares to appear identical. Conscious knowledge cannot simply reach backwards and rewrite every automatic stage of visual processing. Perception follows mechanisms that operate rapidly and largely outside deliberate control. One can know the truth and continue seeing the illusion, a condition with applications extending well beyond geometry. Illusions have helped researchers investigate contrast, sensory adaptation, grouping, constancy, prediction, depth estimation, motion processing, attention, and multisensory integration. They reveal which features the nervous system compares and which assumptions it normally makes. This does not mean perception is fundamentally unreliable. The same mechanisms usually produce successful behavior. Size constancy keeps objects stable across distance. Lightness constancy keeps surfaces recognizable under changing illumination. Grouping turns scattered edges into coherent objects. Prediction allows recognition when signals are incomplete. Illusions are memorable precisely because ordinary perception works so well. A rare mismatch attracts attention against a background of countless successful interpretations. The brain creates experienced reality by following consistent construction rules. Unusual images, sounds, and sensory conflicts expose those rules because the result no longer matches a ruler, light meter, or physical description. An illusion is therefore not simply a trick played upon a defective mind. It is a glimpse of the hidden work that makes the visual world appear effortless, stable, and complete. The body usually feels like the most certain part of experience. A hand seems unquestionably to be your hand. A movement appears to begin because you intended it. Sensations seem to occur within a body that occupies one position and belongs to one continuing self. Yet this feeling is not produced by one single sense. It emerges from the brain combining touch, vision, proprioception, balance, movement, and signals from inside the body. One useful concept is the body schema. This is a largely unconscious representation of the body's size, position, and arrangement. It helps guide posture and movement without requiring constant deliberate thought. When reaching for a cup, the nervous system estimates where the arm begins, how far it can extend, the angle of the wrist, and the position of the fingers. Most of this happens without a detailed visual inspection of each joint. The body image is related but somewhat different. It refers more closely to conscious perceptions, beliefs, and attitudes about the body. A person may have an accurate body schema for movement while holding a distorted or emotionally complicated body image. These are not perfectly separate systems, but the distinction helps explain how the body can be represented in several ways at once. The flexibility of bodily ownership becomes clear in the rubber hand illusion, developed by psychologists Matthew Botvinnik and Jonathan Cohen. A participant places one real hand out of sight while a realistic rubber hand is positioned in view. An experimenter strokes the hidden real hand and the visible rubber hand at the same time and in matching locations. After a short period, many people begin to feel as though the touch is occurring on the rubber hand. Some also report that the artificial hand feels partly like their own. The brain receives visual evidence of the rubber hand being touched, and matching tactile evidence from the hidden real hand. Because the timing and location agree, the nervous system infers that the seen hand is the source of the felt sensation. Ownership has shifted, at least temporarily. The illusion does not usually cause a person to forget that the hand is made of rubber. Conscious knowledge remains intact. Yet the immediate bodily experience changes despite that knowledge. Similar principles can alter the perceived location of the entire body. Neuroscientists Henrik Ehrson and Olaf Blanke have used combinations of visual and tactile information to produce full-body and out-of-body illusions. In some experiments, participants view an image of their body from behind, while touch applied to the real body is visually matched with touch seen on the projected or virtual body. Under suitable conditions, people may feel displaced towards the seen body, or experience the center of the self as located outside its usual position. Vestibular signals can also contribute to unusual experiences of movement, elevation, or self-location. Clinical reports and laboratory studies suggest that disturbances in the integration of vision, touch, proprioception, and balance can sometimes produce the sensation of observing the body from elsewhere. One brain region connected with these processes is the temporoparietal junction. It lies near the meeting point of the temporal and parietal lobes and contributes to integrating information about body position, visual perspective, attention, and spatial location. The temporoparietal junction is not a single control center for the self. Bodily awareness depends on widespread networks. Even so, unusual activity or disruption in this region has been associated with changes in perspective and self-location. Two further aspects of bodily selfhood are ownership and agency. Ownership is the feeling that a body part, sensation, or experience belongs to oneself. Agency is the feeling of causing an action. When you decide to lift a hand and then see and feel it rise, ownership and agency normally agree. The hand feels like yours, and the movement feels self-produced. The nervous system helps create agency by predicting the sensory consequences of its own motor commands. When a movement command is sent to the muscles, a related internal signal may also be produced. This is called an efference copy or corollary discharge. The brain can use this copied information to predict what the movement should feel like and what changes it should create in vision and touch. If the predicted and actual signals match, the action is more likely to be experienced as self-generated. This is one reason it is difficult to tickle yourself effectively. When one hand moves towards the other, the nervous system predicts the location, timing, and intensity of the coming touch. Because the sensation is expected, its impact is reduced. Another person's touch is harder to predict precisely, and therefore remains more surprising and usually more ticklish. The brain has, in effect, ruined its own joke before it begins. Prediction also helps distinguish movements caused by the body from movements in the external world. When the eyes move, the retinal image changes dramatically. Motor-related signals help the brain identify those changes as self-generated rather than concluding that the entire room has suddenly shifted. Bodily selfhood also depends on interoception, the perception of conditions inside the body. Interoceptive signals include information associated with heartbeat, breathing, hunger, thirst, fullness, temperature, blood pressure, inflammation, and the state of internal organs. Much of this regulation remains unconscious, but some signals become feelings such as breathlessness, nausea, tension, calmness, or a pounding heart. The British physiologist Charles Sherrington helped establish distinctions among exteroception, proprioception, and interoception. Exteroception concerns signals arising mainly from outside the body, such as light, sound, and contact with surfaces. Proprioception represents the position and movement of the body. Interoception concerns the internal physiological condition of the organism. Modern researchers, including Antonio Damasio, Hugo Critchley, and Sarah Garfinkel, have explored how interoceptive signals contribute to emotion, decision-making, anxiety, attention, and the feeling of self. The insular cortex is one important region involved in representing internal bodily conditions. Other areas of the brainstem, thalamus, cingulate cortex, and frontal cortex also contribute. Interoception is not always perfectly accurate. A person may notice every change in heartbeat while anxious, yet overlook hunger while deeply absorbed in work. Internal signals are filtered and interpreted just like information from the external senses. Emotion is closely connected with this process. A rapid heartbeat may be experienced as excitement in one setting and fear in another. The physical signal is similar, but context and expectation help determine its meaning. The body that appears in consciousness is therefore both external and internal. Vision shows its shape. Touch defines its surface. Proprioception locates the limbs. The vestibular system provides orientation. Motor predictions distinguish self-produced actions. Interoception supplies a quieter sense of internal condition. None of this proves that the entire self can be reduced to sensory integration. Personal identity also involves memory, language, relationships, values, imagination, and a continuing narrative across time. Even so, the most immediate sense of being someone begins with the feeling of being embodied. The brain continually combines signals into a stable model of a body located here, acting now, and belonging to me. That model is usually so reliable that it feels like a simple fact rather than an active construction. Yet a rubber hand, a mirror, a virtual body, or an unexpected sensory conflict can reveal how flexible it really is. The boundaries of the self feel solid, but the nervous system is quietly redrawing them all the time. Sensory information never arrives in an empty mind. Every sight, sound, smell, taste, and bodily feeling is interpreted within a history of previous experience. The same physical event can therefore mean very different things to different people. A distant siren may be ordinary background noise to one listener and an immediate source of anxiety to another. A particular song may sound pleasant to someone hearing it for the first time, while carrying years of personal significance for someone else. The sensory signal may be shared. Its meaning is not. Memory allows the brain to recognize patterns rather than encountering every moment as entirely new. A face becomes familiar. A voice is identified after only a few words. A place can be recognized from the shape of a street, the echo of a hallway, or the smell of a particular room. Without memory, sensation would provide constantly changing fragments with little continuity. Recognition depends on comparing current information with patterns stored from earlier experience. The hippocampus plays an important role in forming and organizing episodic memories, which concern events experienced at particular times and places. It also contributes to spatial memory and navigation. The hippocampus does not store every memory as a complete recording. Memories are distributed across networks involving many brain regions. Visual features may depend partly on visual areas, sounds on auditory regions, and emotional responses on other circuits. The hippocampus helps connect elements of an experience so that they can later be remembered together. This binding allows the sight of a building, the smell of rain, and the sound of a familiar voice to become parts of one remembered event. Memory is reconstructive rather than perfectly reproductive. When an event is recalled, the brain rebuilds it from stored details, general knowledge, current expectations, and later experience. This is usually useful, but it also means that memories can change, lose detail, or absorb information that was not present originally. The amygdala contributes to learning the emotional and motivational significance of sensory events. It is often described simply as a fear center, but that is too narrow. The amygdala responds to many forms of biological relevance, including threat, reward, novelty, uncertainty, and socially meaningful information. If a neutral sound repeatedly predicts something unpleasant, the nervous system may begin treating that sound as important. Attention shifts towards it more quickly. The body prepares to respond. The memory of the event may also become stronger. Emotion influences perception at several levels. It can guide attention, alter sensitivity, shape interpretation, and affect what is stored in memory. A person walking alone at night may notice small movements and uncertain sounds that would be ignored during a relaxed afternoon. The sensory signals have not necessarily become stronger. Their possible importance has changed. Two useful dimensions for describing emotional experience are affective valence and arousal. Valence concerns whether something feels pleasant, unpleasant, or relatively neutral. Arousal concerns the level of activation or intensity, ranging from quiet and calm to alert and highly energized. A gentle piece of music may be pleasant and low in arousal. A sudden celebration may be pleasant but highly arousing. A dull ache may be unpleasant with moderate arousal, while an unexpected crash may be unpleasant and immediately activating. These dimensions do not capture every detail of emotion, but they help explain how sensory events acquire tone and urgency. Smell provides one of the clearest examples of sensation becoming personal experience. A particular perfume or cooking aroma may evoke a vivid memory of a person, house, holiday, or period of childhood. The molecules themselves do not contain those memories. Their pattern has become connected with earlier experiences through learning. Music can work in a similar way. A sequence of sounds may carry little emotional weight for one listener, while producing comfort, sadness, or excitement in another. Familiar voices also gain significance. The same frequencies and speech patterns that identify a person can signal safety, authority, affection, or tension depending on the history of the relationship. Comfort foods combine taste, smell, texture, warmth, memory, and expectation. Their appeal may depend partly on nutrition and partly on associations with care, routine, family, or familiar surroundings. A bowl of soup is chemically real, but comfort is not one of its measurable ingredients. The neuroscientist Antonio Damasio proposed the somatic marker hypothesis as one account of how bodily and emotional signals influence decision-making. According to this idea, previous experiences become associated with bodily states. When a similar choice appears later, signals linked with those earlier outcomes may help guide attention and behavior before a person has consciously examined every possibility. A subtle feeling of unease may discourage one option. A sense of anticipation may favor another. These bodily markers do not replace reasoning, and the hypothesis remains debated in its details. But it highlights an important point. Decisions are not made by detached thought alone. Internal bodily and emotional signals can shape what seems significant. Perception is also state-dependent. What becomes noticeable and how it is interpreted can change with fatigue, anxiety, hunger, mood, and expectation. When tired, a person may process information more slowly and miss subtle changes. Anxiety can increase attention towards possible threats and encourage threatening interpretations of ambiguous signals. Hunger makes food-related sights and smells more noticeable. A person expecting bad news may interpret a neutral expression differently from someone expecting reassurance. Mood can influence memory as well. Experiences matching the current emotional state may become easier to recall, although these effects vary and should not be treated as rigid rules. The body's needs continually reshape the perceptual landscape. When thirsty, water becomes more important. When cold, signs of warmth stand out. When lonely, social signals may carry greater weight. Experience can also train perception through perceptual learning. Repeated practice changes the ability to detect, distinguish, and organize meaningful patterns. A musician learns to identify small differences in pitch, timing, harmony, and tone. This does not necessarily mean the ears become fundamentally more sensitive to all sound. Attention and neural processing become better tuned to the distinctions that matter within music. A radiologist learns to detect subtle patterns in medical images that an untrained observer may overlook. A birdwatcher identifies species from brief calls, silhouettes, and movements. A perfumer learns to separate components within a complex scent. An athlete becomes sensitive to body position, timing, and the early movements of an opponent. Language learning provides another example. Infants can initially distinguish a wide range of speech sounds. With experience, perception becomes increasingly specialized for the sound categories used in the surrounding language. Distinctions that matter are strengthened, while unfamiliar contrast may become harder to notice. Expertise, therefore, changes the organization of attention and interpretation. It often creates more useful perception rather than simply stronger sensation. Two people can look at the same image or listen to the same recording while extracting very different amounts of information. One notices isolated features. The other recognizes a structured pattern, because years of learning have made that pattern meaningful. Perceived reality is not merely a map of physical surroundings. It is a world filled with familiarity, danger, attraction, comfort, opportunity, and personal significance. Light and sound provide physical signals. Memory identifies what has been encountered before. Emotion assigns urgency and value. Bodily needs determine what matters now. Learning reveals patterns that once remained invisible. The world reaching the senses may be shared, but the world entering experience is shaped by everything the nervous system has lived through. Human beings share the same broad sensory architecture. Most people have eyes that detect a narrow band of light, ears that respond to a limited range of vibration, skin that registers pressure and temperature, and chemical receptors involved in smell and taste. Yet no two sensory worlds are exactly alike. Differences begin at the level of receptors and continue through neural development, health, aging, learning, attention, language, and culture. Even when two people stand in the same room, the physical signals available to them may be interpreted with slightly different sensitivity and significance. Color discrimination varies because the cone photoreceptors and their pigments are not identical in every observer. Some people distinguish very fine differences between shades that others group together. Inherited variations affecting long wavelength or medium wavelength, cone photopigments can make certain reds, greens, browns, and oranges more difficult to separate. Hearing range also differs. Sensitivity to high frequencies commonly declines with age, but the pattern and rate vary greatly. Exposure to loud noise, genetics, illness, and medication can all alter which sounds remain available. One person may notice the faint buzz of an electrical device that another person cannot hear at all. Smell sensitivity can differ by orders of magnitude for particular molecules. A compound may be vivid to one person, faint to another, and effectively odorless to someone whose receptor genes do not respond strongly to it. Taste varies as well. Differences in taste receptor expression can affect sensitivity to bitterness, sweetness, and other qualities. Certain bitter compounds seem extremely intense to some people and relatively mild to others. Pain sensitivity, balance, adaptation, and susceptibility to perceptual illusions also vary. An image that produces a powerful illusion for one observer may have a weaker effect on another. These differences do not imply that one person has correct perception while the other has failed. They reveal normal variation within the same general biological system. A particularly vivid example is synesthesia. Synesthesia is a family of experiences in which stimulation, symbols, or concepts in one domain consistently evoke an additional experience. A person may see particular letters or numbers as having specific colours. Days of the week may occupy fixed positions in space. Musical notes may produce colours, shapes, or textures. These associations are usually automatic and stable over time. The letter A may always appear red, while Tuesday may always occupy a particular location to the left. The added experience is not normally chosen deliberately. The British scientist Francis Galton described synesthetic experiences during the 19th century, including people who visualised numbers and sequences in organised spatial forms. Modern researchers, including Simon Baron-Cohen, Julia Simner, and David Eagleman, have helped study the consistency, development and neural basis of different forms of synesthesia. Synesthesia does not represent one single condition. Its forms differ widely, and proposed explanations include unusual connectivity, altered communication among brain regions, and differences in how concepts activate sensory representations. Variation becomes even clearer when a sensory system is absent from birth. Congenital blindness should not be understood simply as sighted experience with the pictures removed. A person born blind develops ways of representing space, objects, movement, and social surroundings through touch, hearing, proprioception, language, and action. Likewise, congenital deafness is not merely ordinary hearing with the sound turned down. Deaf individuals may organize communication, attention, rhythm, and social information through vision, touch, movement, and signed language. The brain adapts to the information available during development. This ability is part of neuroplasticity, the capacity of neural systems to change with experience, learning, injury, and sensory loss. Cortical regions are not infinitely flexible, but their organization can change. Areas usually associated with vision may respond during tactile or auditory tasks in people who are blind. Regions involved in auditory processing may become engaged differently when communication depends heavily on visual language. These changes do not mean that one sense simply transforms into another. They show that brain regions participate in networks whose functions are shaped partly by the inputs, tasks, and learning available to them. Sensory substitution makes this flexibility especially clear. Neuroscientist Paul Bacchi-Rita pioneered devices that converted visual information into patterns of touch. A camera captured features of the environment, and those features were translated into stimulation across the skin. With practice, users could learn to identify shapes, locations, and movement from the tactile patterns. The skin did not become an eye, and the experience was not necessarily ordinary vision. Instead, the brain learned to extract spatial information from a new channel. Modern sensory substitution systems may translate camera images into sound, distance into vibration, or directional information into patterns on the body. Their success depends on training, device design, and the brain's ability to discover stable relationships within the new input. Echolocation provides another example of perceptual adaptation. Some blind people produce clicks with the mouth and listen to the returning echoes. The timing, loudness, and frequency structure of those echoes can reveal the distance, size, shape, and position of nearby objects. Daniel Kish is one of the best-known practitioners and teachers of human echolocation. Research associated with neuroscientist Mel Goodell and others has examined how experienced echolocators process echoes and represent surrounding space. Echolocation can support remarkable navigation, but it should not be described casually as sight through sound. It provides different information with different resolution and limitations. Its importance lies in showing how attention and learning can extract structure from signals that most people barely notice. Autistic people may also experience sensory information differently, although there is no single autistic sensory profile. Some individuals report heightened sensitivity to sound, light, touch, smell, or movement. Others show reduced sensitivity or seek strong sensory stimulation. A sound that disappears into the background for one person may remain intrusive for another. Bright lighting, overlapping voices, unexpected touch, or certain textures may contribute to sensory overload. Research also suggests that multisensory timing, adaptation, prediction, and the weighting of sensory evidence may differ in some autistic people. These findings vary across individuals and studies, so they should not be turned into a universal explanation of autistic experience. Language and culture add further layers of variation. Languages divide colours, directions, smells, textures, and spatial relationships in different ways. Repeatedly using particular categories can train attention and memory towards distinctions that matter within a community. People who use absolute directions such as north and south routinely may develop strong habits of spatial orientation. Specialists with rich smell vocabularies may identify odours more precisely than speakers whose language rarely labels them. This does not mean language completely determines perception. A person can often perceive differences even without a familiar word for them. Language is better understood as one influence that guides categorization, memory, comparison, and attention. Expertise works in a similar way. A birdwatcher hears species within what others call birdsong. A radiologist sees medically meaningful structure within shades of grey. A perfumer separates a complex fragrance into distinct notes. A musician notices tuning and timing differences that pass unnoticed by an untrained listener. Their receptor organs may not be fundamentally superior. Their brains have become tuned to statistical patterns and meaningful distinctions encountered repeatedly. Developmental history shapes which signals become important, which categories become familiar, and which expectations guide perception. Every nervous system is built through a unique sequence of environments, actions, relationships, illnesses, habits, and discoveries. Jakob von Uexküll used the word Umwelt for the perceptual world available to an organism. The concept applies not only across species but in a gentler way across individuals. Human perceptual worlds overlap enormously. We can usually agree that a light is bright, a surface is rough, a siren is loud, or a room is cold. Shared biology and a shared environment make communication possible. But within that common world are differences in colour, sensitivity, association, attention, memory, and meaning. Every person therefore inhabits a reality that is partly collective and partly private. The same world reaches us, but it does not pass through identical receptors, identical histories, or identical minds. Human senses reveal only a small portion of the physical world. Eyes detect a narrow band of electromagnetic radiation. Ears respond to a limited range of vibration. Skin registers certain forms of pressure, temperature, and damage. Smell and taste sample only molecules that can interact with particular receptors. Yet human knowledge extends far beyond those biological limits. Scientific instruments allow signals that cannot be sensed directly to be detected, measured, and translated into forms that the nervous system can interpret. They do not remove the need for perception. Instead, they create new pathways between otherwise inaccessible events and the human mind. A microscope takes structures too small for the unaided eye and produces an enlarged visual image. An optical telescope gathers far more light than the pupil and can resolve objects that appear as faint points or remain completely invisible without assistance. Other instruments move beyond visible light altogether. Radio telescopes detect electromagnetic waves, with wavelengths far longer than those the eye can register. Infrared telescopes measure radiation associated with cooler objects, warm dust, planets, and distant galaxies. Ultraviolet, X-ray, and gamma-ray observatories record increasingly energetic forms of radiation. A spectrometer separates light or other radiation according to wavelength. The resulting spectrum can reveal chemical composition, temperature, motion, pressure, and magnetic fields. A distant star remains impossibly far away, yet its light carries patterns that allow scientists to identify elements within its atmosphere. Microphones perform a related transformation for sound. They convert pressure variations into electrical signals that can be amplified, recorded, analysed, slowed down, or shifted into a different frequency range. Ultrasound beyond ordinary human hearing can therefore be made audible or displayed visually. Thermal cameras detect infrared radiation and translate differences in temperature into an image. Warm surfaces may be shown as yellow, white, or red, while cooler regions appear blue or dark. Those colours are chosen for clarity. They are not what infrared radiation looks like to an eye, because infrared radiation does not naturally look like anything to the human visual system. Medical scanners extend perception inside the living body. X-ray imaging reveals differences in how tissues absorb energetic radiation. Computed tomography combines many X-ray measurements into cross-sectional images. Magnetic resonance imaging uses strong magnetic fields and radio frequency signals to estimate properties of hydrogen atoms within tissues. These images are not photographs in the ordinary sense. They are reconstructions produced from measurements and mathematical processing. Their brightness, contrast, and colour may be adjusted to emphasize medically useful structures. Particle detectors extend perception even farther from ordinary experience. They reveal charged particles, neutrinos, cosmic rays, and products of high-energy collisions by recording flashes, electrical charges, tracks, or other interactions within specialized materials. A particle detector does not allow a scientist to look directly at a particle as though examining a grain of sand. It records the effects produced when the particle interacts with matter. From those effects, researchers infer properties such as energy, momentum, charge, direction, and identity. Gravitational wave observatories detect something even subtler. Facilities operated by the laser interferometer. Gravitational wave observatory measure tiny changes in distance caused by ripples in space-time passing through Earth. The changes are extraordinarily small, far less than the width of an atomic nucleus across an instrument several kilometers long. Lasers and carefully controlled mirrors convert these minute distortions into measurable interference patterns. Scientists can then transform the data into graphs, models, or even audible signals. The resulting chirp associated with merging black holes is not a sound that travelled through space and entered a microphone. It's a translation of changing gravitational wave frequency into the range of human hearing. Scientific organizations carry out such translations on an enormous scale. The National Aeronautics and Space Administration and the European Space Agency operate missions that detect radiation and particles across the solar system and distant universe. The European Southern Observatory runs major ground-based telescopes that collect visible and infrared light. The Laser Interferometer, Gravitational Wave Observatory converts movements in space-time into data that researchers can inspect and compare. These organizations do not simply collect beautiful pictures. They build calibrated instruments, account for noise, test uncertainty, and preserve information about how measurements were produced. An image from radio astronomy is therefore not direct human sight extended to a longer wavelength. It is a controlled representation of measured radio intensity across different positions in the sky. Computers combine observations, correct for instrument behavior, and assign values to pixels. The final image allows human vision to inspect information that originally arrived in a form the eyes could never detect. Representative color is often used to display this information. Different colors may encode different wavelengths, temperatures, chemical emissions, velocities, or signal strengths. When colours are assigned without attempting to reproduce ordinary visual appearance, the result is often called false colour imaging. The phrase false colour does not mean the data are deceptive. The colours function like a visual language. Blue may represent lower temperature, while red represents higher temperature. Another image may reverse that scheme or use colour to distinguish chemical elements. The meaning depends on the scale and method chosen by the researchers. Without that explanation, an image may be beautiful, but scientifically ambiguous. Technology can also alter sensory information close to the body. A cochlear implant converts sound into patterns of electrical stimulation delivered to the auditory nerve. It does not restore natural hearing in the same way that replacing a camera lens restores focus. The implant provides a simplified patterned input that the brain must learn to interpret. Results vary with age, hearing history, anatomy, device settings, and experience. Many users develop strong speech understanding, while music and complex sound environments may remain more difficult. Retinal implants and related visual prostheses attempt to provide pattern stimulation to surviving parts of the visual system. Current systems do not reproduce ordinary sight in full colour and fine detail. They may offer flashes, rough shapes, boundaries, or information useful for orientation. The nervous system must discover meaning within the unfamiliar signals. Brain-computer interfaces follow the same broad principle. Neural activity can be recorded and translated into commands for a cursor, communication device, robotic limb, or other system. In the opposite direction, stimulation can sometimes provide limited sensory feedback. Haptic technologies translate information into touch through vibration, pressure, or movement. A prosthetic hand may provide signals related to grip force. A navigation device may vibrate on one side of the body to indicate direction. These systems remain technically demanding, and their abilities should not be exaggerated. They demonstrate established forms of communication between neural activity, machines, and sensory experience. But they do not yet provide effortless access to every thought or recreate all natural sensation. Sensory augmentation aims not only to replace missing input, but to provide information humans have never naturally sensed. A wearable device might translate magnetic north into a continuous vibration at one point on the body. Another could convert distance into sound, radiation into touch, or digital information into pattern stimulation. With practice, some users may stop treating each signal as a separate code and begin experiencing it as an intuitive feature of the environment. The technology does not create a new biological receptor, but learning can make the translated information increasingly immediate. Every extension of perception still requires interpretation. Instruments must be calibrated against known standards. Background noise must be distinguished from genuine signals. Measurements carry uncertainty. Computer processing can clarify data, but can also introduce assumptions. Scientific models are needed to connect a pattern on a screen with the event believed to have produced it. A measurement is not reality itself. It is an interaction between reality and an instrument, represented in a form that can be recorded and understood. The same is true of biological sensation. An eye does not contain the external world. It responds to light and constructs visual information. A scientific detector does something comparable, with a different range of signals, and usually with more explicit calibration. Human beings can never step entirely outside representation. Even the most advanced instrument produces numbers, traces, images, or sounds that must enter perception and thought. The scientific response to this limitation is not despair, it is method. Measurements are repeated, instruments are compared. Independent research groups test the same claim using different techniques. Uncertainty is estimated. Predictions are checked against observation. Personal impressions are replaced with procedures that others can examine and reproduce. No instrument provides a complete view of reality. A telescope reveals some signals while missing others. A microscope enlarges a narrow field. A scanner emphasizes particular tissue properties. A particle detector records only events capable of interacting with its materials. But these partial views can be combined. Through microscopes, telescopes, spectrometers, scanners, particle detectors, gravitational wave observatories, and sensory prostheses, human perception reaches beyond the ranges granted by biology. Reality remains larger than any representation of it. Yet instruments, experiments, comparison, and collective reasoning allow those representations to become increasingly precise. The senses create the world as it is experienced. Science extends that world by teaching the senses how to receive signals they were never built to detect. The human senses create experienced reality through detection, selection, comparison, integration, prediction, memory, and interpretation. Physical energy and chemical molecules begin the process. Light reaches the eyes, vibrations enter the ears, molecules stimulate smell and taste receptors. Pressure, temperature, movement, and internal bodily changes activate specialized sensory pathways. Through sensory transduction, these events become patterns of electrical and chemical activity within the nervous system. Those signals are then filtered, compared, and organized. Separate senses are combined into one scene. Attention selects what becomes prominent. Expectation helps resolve uncertainty. Memory supplies recognition, while emotion gives events urgency, comfort, danger, or meaning. The external world remains real and places firm limits on perception. Yet the qualities appearing within consciousness are created by the brain. Colour is the nervous system's response to light. Sound is its interpretation of vibration. Flavour emerges from taste, smell, texture, temperature, irritation, vision, and expectation. Bodily presence is assembled from touch, proprioception, vestibular information, interoception, and predictions about movement. Perceptual illusions reveal these construction rules. They do not prove that the senses are generally deceptive. The same processes that sometimes misjudge a carefully designed image usually allow rapid, reliable action in an uncertain world. Evolution did not provide a complete scientific display of the universe. It produced an actionable world filled with objects, faces, voices, dangers, opportunities, surfaces, and bodily needs. Jakob von Uexküll's concept of the Umwelt reminds us that every species occupies its own sensory environment. Human perceptual worlds overlap closely, yet differences in receptors, experience, learning, attention, age, and memory mean that no two are perfectly identical. Scientific instruments extend perception beyond the natural limits of the eyes, ears, skin, nose, and tongue. Yet microscopes, telescopes, scanners, and detectors still translate measurements into images, sounds, and numbers requiring interpretation. Experienced reality is therefore neither a flawless copy of the universe nor a fiction detached from it. It is a biological model, continually tested and corrected through contact with the world. Every familiar moment carries the result of this hidden collaboration. Hearing rain, seeing moonlight, tasting warm tea, or feeling the weight of a blanket requires matter, energy, sensory organs, neural activity, memory, and consciousness to meet quietly in one experience.